Good morning or afternoon now. Welcome everybody. Let's go ahead and get started. We got a busy day ahead of us. Let's talk first about the exam. I was very, very pleased with the results of the exam overall. They were really, really excellent. The averages are nice and high, and the results were even more than that. Almost a third of the class got A's on the lab exam, for instance. So it was really, really excellent. I was very pleased to see that. The averages are nice and high. The overall grades in the class are over 80%. Those are excellent. And there's, again, no reason these shouldn't continue to move upward. You guys are now getting, again, continuing to gain an understanding of what I'm looking for in my essay questions, on the answers, and the level of detail that is necessary. So you should be learning how to be successful in this class, in this environment. And so I'm very, very pleased with that number, and I would like it to continue to increase. I will, like I've done in the past, even though I finished grading a day or two ago, I posted the grades. I was worried that not everybody necessarily gets the notification. So I have not released the exams yet. I will do it today after class like I usually do for 24 hours. So again, you'll have 24 hours after class today to examine your review and review your exam. There you go. And then if you have any questions, contact me. If you're not able to make it during that time, then you can make arrangements during my office hours to review the exam. We can do that as well. All right, our goal for today is to finish off the respiratory system. We've got some dense material we have to cover, so we want to make sure that we handle that. So it's going to be a very busy physiology lab today, a class today. If there's time, depending on if we finish early like last time, we may start the urinary anatomy, but our brains may be tired after today. Today we've got some dense concepts we need to be talking about. So we'll see how the day goes. We have enough time in the next 3 lectures to cover everything we need for the urinary system. So getting a head start isn't a bad thing because, again, we have a Thursday exam. But if everybody's brain fried and ready for the weekend, I'm not going to push it. We'll see how it goes. We still have a lot of assignments due in this. You have one more respiratory assignment due. That is the PhysioX exercise 7 that has 3 activities due on Tuesday after the weekend. Then you have 2 unit reviews and 2 lab simulations, our Labster and our PhysioX. So that should keep you busy over the next 2 weeks. And as we keep talking about, you have that dreaded Thursday exam. So again, the exam is 2 weeks from today. Meaning you only have 2 more weekends to study this material. So make sure you are using that time wisely between now and the exam. All right, questions on any of that? Excellent. You know how much I love my stunned silences. Spectacular. All righty. So let's talk respiratory system. And in particular, we are going to start with a tiny bit of review of our internal and our external gas exchange. So let's see what we can remember about these things. Starting first, let's go with the external gas exchange first. Someone remind me again of the structures that are involved in external gas exchange. Excellent. The alveoli of the lungs and the pulmonary capillaries. Conversely, we can also talk about the mediums in which that exchange takes place. And because we want to keep them in the same order, what would those mediums be? Although if we want to be in the same order, absolutely. So air in the lungs to the blood in the capillaries, in the pulmonary capillaries. Excellent. Capillaries twice. Excellent. We also talked about gases. This is obviously a gas exchange. What are the gases being exchanged? Oxygen and carbon dioxide. Excellent. Which way does oxygen go in external gas exchange?
From alveoli into pulmonary capillaries.
Excellent. So from the air to the blood, or we could say from the alveoli to alveoli to the pulmonary capillary. Now, Alex, I do want you to be careful about one thing. When this gas exchange takes place, and we'll actually talk about the numbers, remember, air is indeed moving from the air into the blood. Carbon dioxide is indeed moving, as we're about to see, from the capillaries into the alveoli, but oxygen isn't 100% in the air and 0 in the blood. Right, carbon dioxide isn't all in the blood, and there's none of it inside of the alveoli. So there is oxygen and carbon dioxide in both locations, just not equal amounts. And so, as we know, it's those pressure gradients that is going to cause it to move from one location to another. And again, I think we just mentioned it, so I will do it here. From the blood to the air, or from the pulmonary capillary to the alveoli. Excellent. Again, no new information there. We're just writing stuff that we already know. And we can do the same thing for internal gas exchange as well. What are the structures?
Systemic capillaries and tissues.
Excellent. What are... Oops, that's what it did wrong. Got it. All right. What are the mediums?
Blood and interstitial fluid.
Excellent. Of course, what are the gases that are being exchanged? Excellent. Oxygen and carbon dioxide. And with internal gas exchange, which way does oxygen move? From blood into the interstitial fluid. Excellent. And which way does our carbon dioxide move? There you go. Perfect. Excellent. Again, no new information. This is all information we talked about on the first day of our lecture of the respiratory system, but I think it's important to remind us of this because we are going to talk about these processes today. In fact, indeed, we have some pretty pictures that go along with this. Notice here, we see our external gas exchange. Oxygen is leaving the alveolus into the pulmonary capillary. CO2 is leaving the pulmonary capillary into the alveolus. And we'll talk about these processes that are taking place in the blood plasma a little bit later. And then here we have our internal gas exchange. With our internal gas exchange, again, it is between the systemic capillary and the tissue. Oxygen goes from the systemic capillary into the tissue. Carbon dioxide comes from the tissue into the systemic capillary. So here we have, and again, we'll talk about these processes of what's happening in the blood plasma a little bit later. But the key to this is we understand these concepts of internal and external gas exchange. All right. Now, to fully understand how this gas exchange works and the significance of it, we need to talk some more physics. We had so much fun talking physics in the last class. I think it's worthwhile for us to do it again here. And we need to get introduced into a new law. What was the first physics law we learned?
Boyle's law.
Boyle's law. And what did Boyle's law say again?
volume is directly proportional to the pressure.
Inversely proportional. It is inversely proportional to it. Absolutely. Pressure and volume are inversely proportional. Absolutely. Excellent. So our next law that we're going to learn is good old Dalton's law. What Dalton's law states is that when a gas is in a mixture, each individual gas in that mixture exerts its own pressure. Basically, what this means, if I have a box of air, right? I take this air that surrounds me and I put it in a box. What is our air that surrounds us primarily made up of? What are the gases in our air? Nitrogen, carbon dioxide. What else? Oxygen. And there you go, and water vapor. These are the four main gases. There are others, absolutely, but these are the four main gases that make up the air that surround us. And remember, as we talked about, when we are talking about measuring pressure, we talk about the number of times a molecule bounces against the wall. All Dalton's law says is that the number of times that an oxygen bounces against the wall, producing its pressure, is not affected by any of the other gases. So for instance, how many times carbon dioxide bounces against the wall, or nitrogen bounces against the wall, or water vapor bounces against the wall, no molecule affects another molecule. So oxygen's pressure in this box would be the same whether it was in here by itself or in here with all these other gases. What that tells us then is one other important factor, that the total pressure of the mixture of gases is equal to the sum of each individual pressure. So if we want to know the pressure of the air that is around us, all we have to do is take the pressure of nitrogen, carbon dioxide, oxygen, and water vapor, add them together, and that will give us the pressure of our air. And luckily, that's a number we know. What is the total pressure of the air around us?
760.
Yeah, it's atmospheric pressure, absolutely, right? Basically, it's the pressure of the atmosphere. And as of course we know, the pressure of the atmosphere is 760 millimeters of mercury. And that is simply equal to the partial, I'm going to write it out the first times, pressure of nitrogen plus the partial pressure of oxygen plus the partial pressure of carbon dioxide plus the partial pressure of the water. You add all of these partial pressures together, and that will tell you what the pressure of the air around us is. That is Dalton's law. Let's see. No silly questions except the question not asked. Great question. So the question was, when we talk about the pressure as counting the number of times it bounces against the wall, like what kind of timeline are we talking about? Are we talking about per minute or per second or something along that? No, we really aren't talking about it in that terms. It's more like the average rate over time. And really the way it's measured, if you noticed, is by the energy it produces, the energy that that bouncing on the wall produces. Because the millimeters of mercury basically means how far can it move mercury, right? If you had a tube where you had a little bit of mercury in there, this gas bouncing on it off the walls produces an energy, and that energy is enough to force that mercury to a certain distance. up that tube. And so really, it is the number of times that it counts on the wall, but really what that's doing is that's producing an energy. And basically it's that energy, that kinetic energy moving the little bubble of mercury. That is what we measure. And that's why we use the millimeters of mercury. It's really a measurement of energy that these gases are moving with. Right? Because the actual number of times these things are bouncing against the wall in a second of time is, you know, gajillion or something crazy like that. They're moving along. All right. Let's clear this and write this out again. So the partial pressure of our gas is the sum of all, the total pressure of our gas is the sum of all the partial pressures. And in air, there are 4 primary gases that make up the air that surrounds us. In order of greatest to least, there is nitrogen, which makes up almost 80%. Oxygen, that makes up most of the rest. And notice there is a tiny bit, about 0.5% of water, 0.04% of carbon dioxide. Now, if we know the percentages of the gas in the air, and we know the atmospheric pressure, we can actually calculate the individual partial pressures of the gases. How would you do that? How could we calculate? Multiply, absolutely. You take the total pressure, right, which in this case is 760 millimeters of mercury. And from that, you do what?
Multiply the percentage.
Okay, excellent. You multiply it times the percentage. But remember, we have to do it as a percentage. So that would be 0. Let's take nitrogen, for instance, 79. Right, so you take it by the percentage of the partial pressure. And as a result of that, there and there. And as a result of that, what would the partial pressure of nitrogen equal? Is it 600 exactly? Perfect. Excellent. Let's do the next one. What is the partial pressure of oxygen? Partial pressure, oxygen would of course be 760 times 21, and that would equal, oops, hold on, 0.60. Excellent, 159.6, which we can round up to 160 millimeters of mercury. Excellent. What is the partial pressure of water? 760 times 0.005, which equals 3.8 millimeters of mercury, and our partial pressure of carbon dioxide, which would equal 760 times 0.04. Oops, hold on. 0.0004. 3 zeros. Thank you. And that equals, excellent, 0.304 millimeters of mercury. Excellent. And just that simply, we can figure out the individual partial pressures of the air that surrounds us. Now, notice one more thing. This only helps us at atmospheric pressure, right? At sea level, where it's 760. So again, if you'll write those numbers down, because those numbers are going to be important, we'll come back to them. We figured out the partial pressure of oxygen. We did that, and it's 160 millimeters of mercury. However, that's only at sea level. If instead we go up to 10,000 feet, the atmospheric pressure becomes much, much less. And so the partial pressure of oxygen becomes much, much less, about 110 millimeters of mercury at 1,000 feet, all right? And if you're in that airplane flying at 50,000 feet in the air, then the partial pressure of oxygen is only 18 in the air that surrounds us, all right? That's why the windows in airplanes don't roll down. Yeah, it'd be a lot of fun to have the wind blowing through your ears, right? But would you be able to breathe at that elevation? No, as we're going to see, 10 millimeters of mercury of, 18 millimeters of mercury of oxygen is going to be, impossible to survive at, which is also why most mountain climbers, right, when they're going 20, 30,000 feet, require the assistance of additional oxygen. They bring oxygen tanks with them to help them to deal with the decrease in oxygen at the increased altitudes. All right. Are we comfortable with this? Because this is just our starting point. If this doesn't make sense, it's just going to get worse. All right, excellent. There is one last law we need to understand, because notice as we put out those numbers for the pressures, clearly what gas has the highest pressure?
Nitrogen.
Nitrogen, absolutely. So why doesn't nitrogen have, well, yeah, all of them change at different altitudes, absolutely. So all the gases, nitrogen, oxygen, carbon dioxide, and water would all change at higher altitudes in the same way. They would all decrease as well because the overall pressure in the air surrounding them would decrease as well. Nitrogen clearly has the highest partial pressure. It makes up the largest part of the air around us. But notice when we talk about gas exchange, we don't really talk about nitrogen. And that question was actually asked in the last class as well. And we kind of left that answer hanging. The reason for that is a third law. That third law is called Henry's law. Henry's law basically states that there are two factors that determine how much of a gas you can get into a watery solution. All right. Basically, this is what Henry's law states. How much gas can we dissolve in a liquid? And there are two main factors that determine that. The first factor is the partial pressure. The higher the partial pressure, the more of a gas we can get into solution. So if, for instance, we doubled the partial pressure of oxygen, would we be able to get more oxygen into our blood? Yeah, absolutely. Right. We do that with hyperbaric chambers all the time. We put a bunch of oxygen in there, or rather than having you breathe the air around you, we put one of those tubes in your nose and feed you pure oxygen. And so by adding more oxygen, by increasing the partial pressure of oxygen in the air, we can get a lot more of that gas into solution. Well, doesn't that mean that nitrogen with such a high partial pressure should be able to get a lot of gas into our blood? Yeah, you would think it would, but there is a second factor. And that second factor is what is known as, there you go, the solubility. Each gas has its own constant rate of solubility. meaning some gases are more soluble and some gases are less. Now, do you think you can change the solubility factor of a gas?
No.
No, of course not, right? This one here cannot be changed. But what about the first one? Do you think we can change this first factor?
Yes.
Yeah. And so it is by changing this first that we're able to get more and more gas into solution. You probably have never thought of Henry's law before. It's quite possible, but you have all experienced this, because I'm fairly certain every single person in this class has opened up a can of soda, or if it's even better, in a bottle of soda, because that bottle is clear. What happens when you open a bottle of soda? Come on, I know at least one of you have opened it before. Oh, okay, so release it in what form? How do you know you release CO2? Bubbles. There you go. Absolutely. You see the bubbles. That Coke is sitting there on the counter with the cap on, and all you see is a black liquid. But the second you pop the top, all these bubbles appear in solution. Yeah, absolutely. You can hear the bubbles fizzing. You can hear the gas being released. Absolutely. And again, as you guys correctly have surmised, that gas is carbon dioxide. That is exactly what they do. What happens is they take a can of soda and then they pump a massive amount of carbon dioxide into it. And as they pump that massive amount of carbon dioxide into it, we get a very high partial pressure. Oops. And that high partial pressure forces that gas into solution. And it dissolves in that liquid. But then you pop the top, and hopefully there's not that much space in between the top of your soda and your soda in the can, but you get the idea. When you pop the top, now that gas is able to leave, because after all, we know that the gas in here is going to move from a high concentration to a very low concentration. In fact, what did we say the partial pressure of carbon dioxide was in the air that surrounds us? That was the percentage. What was the partial pressure? There you go. 0.304%. Oops, sorry, not percent. Millimeters of mercury. All right. That is our partial pressure. Let's say, for argument's sake, just to keep the numbers simple, let's say that here inside of the can, when they put it in here, let's say they made it 300 millimeters of mercury. Now, obviously, it's not going to be that high, but let's just do that because the math would be easy that way. All right. So we started at 300 millimeters of mercury in here. We popped the top. It was 300 in here, 0.3 outside. So of course, as you know, gas is going to move. Our carbon dioxide moves to reach equilibrium. It leaves the liquid, it leaves the can until it reaches equilibrium. And if it's 300 inside the can and 0.3 outside in the world, then the halfway point is somewhere in between, about 150 millimeters of mercury, right? Is that what happens? Inside the can, it becomes 150 millimeters of mercury, and in the room around you, carbon dioxide is 150 millimeters of mercury? No. Isn't that how equilibrium works? We get the halfway point, do the math.
No.
Well, that is how we get an average, right? So then why is, it's not so much that it's a slower process, right? Because I don't like to drink mine right away. Maybe I like to let mine sit for 2 hours before I drink it, right? Is it still bubbly after 2 hours?
No.
No, why not?
Carbon dioxide leave.
Yeah, it's reached equilibrium with the room around us. And wouldn't that equilibrium be 150 millimeters of mercury? Well, it does dissipate into the air, but again, that's how averages work. It goes from a high level to a low level, and we meet in between. It's like how we figure out the averages on the exam. On the exams, this last exam, one person got 100%, 20 people got 5%. And so the average in between, well, let's say 10. 5 people got 10%, one person got 90%. The average is 45. So the average in the class was 45 because you take 100, you add 10 to it, you get 110, you divide it in half. Oh, I'm sorry, it's 55. So the average for the class was 55, right? Is that how that works?
No.
No. Why not? That's the average between 100 and 10. You guys are overthinking this. Why doesn't that work?
Equilibrium must be reached with the air outside.
Right. It's got to reach equal. Okay. But here's what you're not gasping. Okay. Let's think of it this way. Let's take it a step back. Let's draw one more beaker. Let's go back to 430. I have a beaker that on one side is 90% glucose. And on the other side of the beaker, it is 10% glucose. All right, this beaker is filled with water. And then I poke holes in my diaphragm in between. And what happens in this case?
Go balance out.
And what will the balancing part, what will equilibrium be at this point? How much glucose will be on each side of this beaker?
50.
50. So it'll be 50 on this side, and it'll be 50 on this side. Right? At that point, again, it's not that there's no net move. I mean, it's not that there's no more movement, but no net movement at that point. It's reached equilibrium. So why isn't the halfway point here 150? What's the difference between these two conditions? Between a beaker, where we've got the two sides, and between my can of soda and the room I'm sitting in?
Volume?
Exactly. All right. Here, it's equal because we have equal amounts of water on both sides. Is there the same amount of gas inside of my can as there is in the room that surrounds me? No, of course not. If I take a bucket of boiling water and I pour it into the ocean, which is, freezing water, does it suddenly become nice and warm and toasty as a result of that?
No.
No, because I have such a small volume that I'm adding to the overall. Does the overall really change? If I took a bucket of boiling, scalding hot water and I poured it into the ocean, would the temperature of the ocean change?
No.
No. So if I open a can of soda and release a can of soda's worth of 300 millimeters of mercury carbon dioxide, is that going to change the overall carbon dioxide level in the room?
No.
No. So when I open this can, what actually happens, as you guys have already mentioned, is that the gas is going to leave. Gas will continue to leave until the partial pressure of carbon dioxide inside the can is equal to the outside world. And of course, in this case, it's going to be 0.3 0.304 millimeters of mercury. Point. At which part, what would I consider my can of soda to be? Like I said, do you like to let your soda sit for an hour? No, it's going to be flat. All the bubbles are going to be gone. All the carbon dioxide is going to be gone. You guys all have absolutely the right idea. The reason it doesn't reach that equal point in between is because there's so little gas in that can. Then when I pop it open, we don't reach equilibrium in between the two. The much larger volume of my room drives the can to equalize it. That's how equilibrium is going to work on this. Does that make sense?
Yes.
Excellent. So, like we said, two factors influence how much of a gas we can get into solution: partial pressure of the gas and the solubility. Solubility we can't change. And that's the thing with nitrogen. Nitrogen has an incredibly low solubility. So even though there's a lot of it in the air, it doesn't like to go into water. Right? However, our carbon dioxide, for instance, we put that pressure on it, we get more of the bubbles into the solution. Right? That unopened can of soda. That is correct. If you were in a room that had the same amount of CO2 as was inside of a can of soda, when it opened, the CO2 would stay in the soda, but guess what? It would still taste flat because you wouldn't have the, so you would taste the carbon dioxide in there. You'd get that carbon dioxide taste for it, but you wouldn't get the bubbles because since it would be at equilibrium, the bubbles would stay dissolved in the soda. Great question. I like that question. Of course, also, if you were in a room with that much CO2, you'd probably die, but that's beside the point. All right, but we can change the partial pressure of a gas. We open that and then it leaves. Changing to the room, changing to the environment. And this brings us back to nitrogen. Again, this brings us back to the question from the last class where we talked about scuba divers. The same way that as you go up in elevation, pressure goes down. As you swim deeper and deeper in water, pressure goes up, and it goes up pretty dramatically. So what happens is you go scuba diving, and so they take a big, huge tank, and basically they pump the air that surrounds you into that tank. So that tank has the oxygen, the carbon dioxide, the water vapor, and the nitrogen that is in the air that is around us. Then when you're under the water scuba diving, the increased pressure of the environment around you starts to force some of that nitrogen into your blood. And that nitrogen, as it's being forced into the blood, produces a condition we call nitrogen narcosis. You start to get a little lightheaded. You start to get a little euphoric, feeling a little drunk as a result of it. So that nitrogen necrosis can occur. Of course, if you don't feel well and you come up too rapidly, then just like popping that can of soda where you change the pressure dramatically, those nitrogen bubbles are going to come out of solution. As they come out of solution, they can cause pain and discomfort in the joints. It can cause respiratory problems and things along those lines. All right? Instead, you need to bring the pressure up more slowly. As you bring the pressure up more slowly or down more slowly, then you're able to exhale that nitrogen, get it out. But if you go too fast, it causes nitrogen to come out, causing a condition we call a decompression sickness. Exactly, decompression sickness, which is also commonly referred to as the bends. Exactly. And of course, if you're dealing with the bends, what do they do for you? Do you know if you've got decompression sickness, what they can do to help you? You know, you guys know. They put them in a decompression chamber where they put the pressure back up high, and that pressure pushes the nitrogen back into solution. And then over the course of a couple hours, they slowly decrease the pressure. And as they slowly decrease the pressure, that nitrogen is able to come out of solution safely just from breathing, and you don't get that decompression sickness. We do the opposite with things like carbon monoxide poisoning. If you have carbon monoxide, as we talked about, that carbon monoxide likes to bind to the heme of our oxygen, and it binds with a higher affinity than oxygen does. So when it gets on there, it doesn't like to let go. So what do? We put you in a hyperbaric chamber. We fill it with a tremendous amount of oxygen, and we dramatically increase the pressure. And that increased pressure is able to force the oxygen into the blood. And it can finally push that carbon monoxide off. Right? Hyperbaric chambers were first used for this types of things, but now you hear about them being used all the time for all sorts of other things. Right? Michael Jackson slept in a hyperbaric chamber because he felt it helped him to regenerate more rapidly from the activities of the day from doing that. Kobe Bryant, right? When he was recovering from knee injury, he went and had a whole bunch of, he went to Europe and had several non-FDA approved surgeries done on his knee, some stem cell research, procedures and things like that. And they also put him in a hyperbaric chamber because, as you know, if we can get more oxygen into it, we can hopefully get it to heal more quickly. And so more and more athletes are using hyperbaric chambers for things along those lines as well. Right. So again, what a decompression chamber would be is it would be like taking that, well, yeah, sort of like that. Yes, you would want to try to get the gas back into solution so that it can come out more slowly instead of all coming out at once. All right. So that's our goal. Our goal is to passively move those gases. And now we understand how we make those factors, right? We want to have the biggest partial pressure as possible, because now we know the change in partial pressure is what drives the diffusion of the gases. We know we have a super tiny, thin respiratory membrane with a huge surface area. And so with those, we can try to get as most efficient as possible gas exchange that we possibly can get. All right. And again, also gases are small and nonpolar, so they're lipid soluble. So do we need special transporters or special channels to allow the gases into or out of the cell to have them cross the plasma membranes? Or can we just use simple diffusion? Remember, simple diffusion is when the gas passes without any type of channel, without any type of protein, without any type of assistance. All right. So great question. The gas bottles that they use when you're doing scuba diving typically just are compressed air. So all they do is they take the surrounding air that's around you and they cram it into a bottle. Now, if you are doing long-term scuba diving or you are doing very deep scuba diving, then they will use a special mixture of gases that will have less oxygen, will have less nitrogen in it. But if you just go down to Monterey Bay and take a 3-hour scuba diving lesson, they're just taking their tanks and taking the surrounding air that's around them and putting the surrounding air into there. So the partial pressures of the 4 gases are the same inside that tank as they are around you. And that's why when you go into the water, some of that nitrogen is forced into the blood.
Okay, thank you.
Yep. All right. Any other questions on that? All right, we need to do some math now and talk some numbers. This, I want to make sure we have time to go over properly. So even though it's a teeny bit early, let's go ahead and take our first break here. We'll take our first break. We'll take a 15-minute break, come back at 1:10. And at 1:10, we will start learning some numbers. All right, so any questions on either of our two laws that we just talked about today, Dalton's or Henry's, before we take our first break? All right, I'll see you in 15 minutes then. All right, excellent. So good thing that nobody answered me. Oh, someone finally did. Excellent, because it reminded me that I needed to record this. So we are going over the numbers. We're going to do it once with me just drawing it on the board. So for this, I really want you to sit back and just watch and think and listen. And then we'll look through it at all the pretty pictures and all the pretty words and make sure it makes sense, because there are some tricks here. And so I want to make sure that you catch those tricks. Absolutely correct, Alexa. The very first pressure is, of course, 160 millimeters of mercury, which is the partial pressure of oxygen out here in the environment surrounding us. And remind me again what the partial pressure of carbon dioxide in the air that surrounds us is. There we go. 0.304 millimeters of mercury. Oops. Mercury. Perfect. Excellent. So those are partial pressures we already know. That's our starting point. So there are some new pressures we need to talk about. Now let's start again, easy. We know, of course, when we do this atmospheric air comes down into the lungs. Let's not use blue. Comes down into the lungs. So of course, here in the alveolus, we are going to have a partial pressure of oxygen and a partial pressure of CO2. Excellent. And of course, after you breathe in that air, what is the partial pressure of oxygen? What is the partial pressure of CO2 going to be when you just take that air from up here and move it into your lungs?
Same.
Exactly. Your partial pressure of oxygen is going to be 104 millimeters of mercury, and your partial pressure of carbon dioxide is going to be 40 millimeters of mercury. Excellent. All right. Questions on that?
It's not the same.
Oh, you're right. Those aren't the same. Why aren't they the same?
Is it a smaller area?
Okay, true. But if I grab a small amount of this air and I put it in a bubble, is that really going to change the partial pressures of them?
No.
Well, isn't that what I'm doing in my lungs? I'm taking a small amount of air in here and putting it inside of this completely empty bubble, filling it with the gases that weren't there before. Because that's how we breathe. When you breathe out, your lungs become a vacuum that have absolutely nothing inside of it, so that when you fill it with air, you were just taking the air that's inside and putting it in, capturing it in a bottle. Is that what we do?
No, there's a residual air.
Excellent. And if only that residual air had a name. There is air. When we breathe, there is still air in the lungs, in the alveoli. What was the name of that air that is still in the lungs even after we do a normal exhalation? If only we had learned them in the last class. They're close. You get partial credit for that. It is the now we can just put those two together. Functional residual capacity. Excellent. And I know I told you you didn't have to memorize the numbers of that, but if you're looking back at your notes, what did we say the functional residual capacity was on average? Well, okay, let's ask the important question first. How do you calculate it? How do you calculate the functional residual capacity? Because that's definitely something we need to know for the exam.
ERV and residual volume.
Excellent. ERV and RV, of course, we need to know what those mean. Expiratory reserve volume and the residual volume, absolutely. And what was the functional residual capacity on average? About 2,400 milliliters. Absolutely, right? Our expiratory reserve volume was around 1,200 milliliters, and our residual volume was about 1,200 milliliters. Now, when we take a normal breath, what was that average volume of a normal breath, that tidal volume? Only about 500 milliliters. Now, again, you don't have to memorize these two numbers, but what I want you to appreciate is what we're doing. You have 5 times as much air in your lungs as what you take in with a normal breath. So if you have, like I said, if you have 5 liters of water and you add 1 liter of boiling water to it, is all the water going to be boiling? No. And that's what happens here. Remember, this functional residual capacity can can participate in gas exchange. So it's going to have a low amount of oxygen. It's going to have a high amount of carbon dioxide. So when we take this air that has a lot of oxygen and very little CO2, and we mix them together, so basically we mix the two volumes together. We mix the functional residual volume plus the tidal volume. When we put those things together, this is the partial pressures that we end up with in our alveolus. So when we mix those two volumes together, it has a little less than the air, oxygen than the air that surrounds us, and a lot more carbon dioxide. Does that make sense why it's different? The partial pressures in the alveolus are different from the atmospheric pressures. Does that make sense? Remember, I take stunned silences as yeses, which mean that you totally understand the information and I can make it harder. So yes, that makes sense? All right, excellent. All right, so now we know what's going on in the alveolus. We know the partial pressures in the alveolus. So now we need to talk about what the partial pressures are in what we've talked about before as our oxygen poor blood. All right, we know that this systemic vein is carrying blood back to the right ventricle, pardon me, the right atrium. And we've up to this point called it oxygen poor blood. But we've never actually calculated what that means. And so let's give it some partial pressures. It turns out that in our quote unquote oxygen poor blood, the partial pressure of oxygen is actually equal to 40 millimeters of mercury. Remember, one of my pet peeves that I had on the very first day of class is that we have no such thing as deoxygenated blood. Deoxygenated means you have no oxygen. Clearly, that is not the case. Here we have oxygen. Not a lot of it, but absolutely we have some oxygen. All right. And the partial pressure of CO2 is about 45 millimeters of mercury. So here in the right atrium, our partial pressure of oxygen is 40 millimeters of mercury, and the partial pressure of CO2 is 45 millimeters of mercury. Questions on that? Well, the talk about a can of soda made me want to go get a can of soda. All right, excellent. Well, that and the caffeine. All right, stunned silences, excellent. That means we understand that. Where does this blood in the right atrium go?
Right ventricle.
Right ventricle, excellent. So this is going to go into the right ventricle. And from the right ventricle, where is it going to go?
Pulmonary trunk.
Pulmonary trunk, pulmonary artery, ultimately leading to a pulmonary capillary. Excellent. So absolutely, these are the pulmonary arteries, which include the pulmonary trunk, right? And everything else that goes along with it coming out of that right ventricle. We know that. So let's actually do that. Trunk/arteries. Excellent. So great question. Someone asked, is there a difference for each person? Not much. There may be minor fluctuations, but this is not like some of the other numbers we've talked about before, like height and things along those lines. These numbers matter. In fact, these numbers you are going to memorize because these are numbers you have to know. So these numbers are the same for everybody. Excellent. So these come to our pulmonary capillary. And as it enters the pulmonary capillary, have these values changed at all? Is there going to be any change in the partial pressures of oxygen and CO2 between the right ventricle and the pulmonary capillaries?
Maybe not.
No, of course not. Remember, we know exchange only takes place in capillaries. So we've got a partial pressure of oxygen, that is equal to 40 millimeters of mercury as it enters. And our partial pressure of CO2 is 45 millimeters of mercury. Excellent. Now, as it enters into our capillary, we know gas exchange is going to take place in this pulmonary capillary. All right. So actually, I'm going to cheat, move this right outside so that I have some room to play in here. Now, oxygen. Which way does it move?
Air to blood.
Excellent. Excellent. So we know oxygen, and for oxygen, we'll go ahead and use red because it's convenient. We know oxygen is going to move, not form, but from the air to the blood. Until what happens? What's always is the goal of these types of movements? To reach equilibrium. What is equilibrium going to be in this case? We have 104 in the alveolus, and we have 40 in our capillary. I'm glad that nobody jumped to say 72. Why is 72 not the right answer? He says drinking from a very small can of soda.
Yeah, 104.
Exactly. It's going to be 104. Why?
Because it's the outside pressure.
Not a bad guess. It isn't so much that it's the outside pressure, but it's the size of the alveolus. Remember, we saw what that alveolus looked like, made up of those simple squamous type 1 pneumocytes. And remember when we saw that big, massive alveolus, on the corners of it, we saw the tiny little capillaries that drape around it. Remember, capillaries are so tiny that our red blood cells have to line up single file to get through it. So the volume of our alveolus is much, much larger than the volume of the capillary. And when you have those bigger volumes are going to drive the equilibrium. So absolutely, at this point, it's going to drive it until it reaches equilibrium with the alveolus. And in that case, that's going to be 104 millimeters of mercury. So when that gas leaves, the partial pressure of oxygen is equal to 104 millimeters of mercury as it is leaving the capillary. So our oxygen has moved from the air to the blood. And now we call it oxygen rich. And we know it's going to leave. All right. Excellent. Let's do the same thing. And actually what I'm going to do here is I'm going to cheat. Let's move this over here. Move that like that. Grab this and move that there. And then we can do the same thing over here with carbon dioxide. Which way does it move?
Blood to air.
Excellent. And it's going to move from blood to air until it reaches equilibrium. And of course, equilibrium with the alveolus. So in this case, what is that partial pressure going to be?
45.
Well, remember, it's coming in at 45. What's the partial pressure in the alveolus? 40 millimeters of mercury. Excellent. So our carbon dioxide, and we'll use green for that, is going to move that way. Moves into the alveolus till it's equal. And so as it leaves, the partial pressure of CO2 is equal to 40 millimeters of mercury. There. And there. Right? When you open that can of soda, the can changes, not the room. And so it's the same thing here. The alveolus is the room. The blood capillary, the pulmonary capillary is the can. So the gases in the capillary change to what the partial pressures are in the alveolus. And now we have oxygen-rich blood, because that's what we talked about from the very first class. Oxygen-rich blood leaves the lungs and comes back. And now we know oxygen-rich blood is 104 millimeters of mercury. And we know our oxygen poor is 40 millimeters of mercury. And that's a pretty big swing. But look at our CO2 levels. What is our CO2 level swinging back and forth between?
Only 5 millimeters difference.
Yeah, it's only a 5 millimeter difference. Notice the goal isn't to get rid of all of the CO2. In fact, after doing the lab that was due today, why wouldn't you want to get rid of all of the CO2 that was in your blood? Right, because that CO2 in your blood is making the blood acidic. It's maintaining the pH. If you were to get rid of all of that CO2, your blood would become very basic, very alkaline as a result of that. And that would be a very, very bad thing. So notice we want to lower it, but we don't want to get rid of it entirely. Again, we keep talking about, when a lot of laymen talk about respiration, they think it's all about oxygen. And there are big fluctuations of oxygen, but the small changes in CO2, being the sophisticated A&P student that you are, shows you that there's a much more narrow range of carbon dioxide levels. And so that carbon dioxide level is much more significant, much more important for affecting the function of the body. All right, excellent. We have completed our external gas exchange. We know which way the gases flow, and we now know the partial pressures associated with it. So now we know that this oxygen-rich blood comes back through the pulmonary veins. And it goes back where?
Left ventricle.
Excellent. It's going to go into the left ventricle. Right. So here into the left ventricle.
It's atrium, right?
I'm sorry?
It's an atrium. In this case, it's atrium.
Oh, sorry. I'm sorry. Yes, yes. Atrium. I'm sorry. Thank you. Into the left atrium. I apologize. Thank you for catching that. Excellent. And now, of course, this is our partial pressures of our oxygen-rich blood. And again, like I mentioned before, no capillaries in the way there. So our partial pressures of oxygen and CO2 going to change as they go back from the pulmonary veins back to the heart?
No.
Of course not. So our partial pressure of CO2 is 40 millimeters of mercury, and our partial pressure of oxygen is 100 millimeters of mercury. Excellent. All right. Questions on that? What do you mean? It's 100 millimeters of mercury.
It's because of bronchial circulation, right?
Ah, exactly. There you go. Noticed, absolutely. Remember, our oxygen level has dropped. So again, note, our partial pressure of oxygen is lower. And that does seem odd, but remember, you get it on the head. Remember, on the way back, our bronchial veins feed some oxygen poor blood into the pulmonary veins, and that slightly drops the oxygen level.
That doesn't change the CO2 level though?
Remember, it's a relatively small amount of blood that's going in. And so notice so little blood that our oxygen only dropped 4 millimeters of mercury. Remember, oxygen poor blood is usually around 40. So the difference between 40 and 45 isn't significant enough where the small amount we're putting in doesn't change the partial pressure of the carbon dioxide, but it is enough to drop the oxygen levels just the tiniest bit. So it just slightly drops the oxygen levels, but it's not enough blood to affect the carbon dioxide levels. All right. Excellent. Where does this blood go from the left atrium?
Left ventricle.
Into the left ventricle. And where does it go from the left ventricle?
To the systemic
To
circulation.
the systemic capillary. Excellent. All right. Now, are our partial pressures going to change again by the time that they reach here to the systemic capillary?
Should not.
Now they don't, exactly. So indeed, the partial pressure of oxygen as it enters, let's put it here. As it enters the systemic capillary, the partial pressure of oxygen is 100 millimeters of mercury, and the partial pressure of CO2 is 40 millimeters of mercury. Excellent. All right. So we know that's what it is coming in to our systemic capillary. So now we might need to talk about what's happening here in our tissues. Our tissues, let's say that our tissues at rest. When a tissue is at rest, does it still need to metabolize? Does it still need to make ATP? Does it still need to be doing maintenance type things?
Yes.
Absolutely. It needs to be doing all those things. So it does work, but not a lot of work. And of course, part of that work is going to be involving producing ATP. And as we know from our cellular respiration, what do you need to make ATP?
Oxygen.
Oxygen. And what do you produce as a waste product from producing ATP?
Carbon dioxide.
Excellent. So while this is at rest, the partial pressure of oxygen is going to be equal to 40 millimeters of mercury at rest. And if it was active, would the number go up or would the number go down?
Down.
Down. So it'd be lower if active. Our partial pressure of CO2 in this resting tissue is 45 millimeters of mercury at rest. And would this one go up or down if active?
Up.
It would be higher if active, absolutely. So, but we'll assume this tissue is at rest. So this tissue is at rest. The partial pressure of oxygen is 40 millimeters of mercury. The partial pressure of carbon dioxide is 45. So let's talk about what happens to oxygen inside this systemic capillary. What direction does it move? Which way does oxygen move in a systemic capillary? From the capillary to the tissue, or let's keep it consistent with what we did for the external gas exchange, from the blood to the interstitial fluid. Excellent. Its goal is to reach equilibrium. And now we know the trick. Is the volume of our capillary or the volume of your muscle in your muscle tissue going to be bigger?
No.
Which one's bigger? The tissue bigger or the systemic capillary bigger? Which one's bigger?
Tissue.
Tissues are definitely bigger, absolutely. So in this case, oxygen is going to move out of the capillary into the interstitial fluid until it reaches an equilibrium point, which at rest would be, what would the partial pressure of oxygen be at the end of this process?
40.
40 millimeters of mercury. Excellent. And our oxygen is moving into the interstitial fluid. Of course, what way does our carbon dioxide move?
From interstitial fluid to the blood.
Excellent. Out of the interstitial fluid into the blood. Absolutely. So for carbon dioxide, it moves from the interstitial fluid to the blood. Again, to reach equilibrium, which in this case would be a partial pressure of CO2 of what?
45.
45 or more. Remember, this can be 45 or more. It can be 40 or less, but again, at rest, we're going to go with 45 as the number. And so notice as the blood leaves, we now have a partial pressure of oxygen that is equal to 40 millimeters of mercury. And oops, let's put it on the same line. And a partial pressure of CO2 that is equal to 45 millimeters of mercury, which, if you notice, is where we started. Because this blood, when it gets back to the heart, doesn't change. 40 of oxygen, 45 of CO2, and then it's moved to the capillaries, where it loads up on oxygen, gets rid of some CO2, and the process continues. Around and around and around she goes.
This exchange occurs at once or by the way, like until it reaches the right atrium?
Great question. So here is the answer. It takes less than a second for your blood to pass through, for instance, the pulmonary capillary. And this gas exchange occurs in about the first quarter of that, quarter of a second, something crazy like that. I actually have a graph that shows that. We will actually look at this in just a second.
Okay.
But it happens incredibly fast because remember, huge surface areas, very thin membrane, strong gradients, especially for oxygen. So the transfer happens really, really quickly. I'm actually not hating this drawing that I've done. It's not bad, but I will say that your book does do a nice job of showing this as well. And there's a little prettier than mine, but you'll notice it shows the exact same thing. Here is our oxygen poor blood as it leaves the tissue, comes back to the right atrium, and then leaves out the ventricle to the right ventricle to go to the lungs. Notice the air that we breathe in has 160 millimeters of mercury of oxygen and only 0.3 millimeters of oxygen of CO2. But remember, in the alveoli, this inspired air is mixing with that. So let's actually put that there to remind us. This is going to be too big. Mixes with the functional reserve volume. And when it mixes with the functional reserve volume, the partial pressures of oxygen in the alveolus is 104 millimeters of mercury. The partial pressure of CO2 in the alveolus is 40 millimeters of mercury. Gas exchange takes place. Oxygen enters into the blood. CO2 leaves the blood. And as the blood leaves, it is now equal to what did I say? 45. I'm sorry. Yes, 40 CO2 in the blood, 104 in it as it leaves the capillary. As it leaves the capillary, leaves the lungs. But remember, when it gets to the heart, the partial pressure of oxygen is now 100 millimeters of mercury, whereas the partial pressure of CO2 is still 40. And again, remind me, why is there this change?
Because of bronchial circulation.
I like that. Let's say it just that way, circulation. Yeah, really, it's the bronchial veins that are feeding it in there, but I like that. Do the bronchial. And again, bronchial, not bronchiole. Make sure you don't make that spelling error when you are taking the test. This goes to the tissues, where the tissues are either at rest or they're active, right? If they're at rest, then they are going to, again, be able to, they're going to be at about 40 millimeters of mercury, and they're going to take on a bunch of oxygen. And then the CO2 is given off. If they're more active and oxygen levels are lower, then they're going to take even more oxygen from the blood. That's the beauty of this process. The more active it is, the lower the oxygen levels, the more oxygen we can take from the blood. The higher our CO2 levels are, the more CO2 we can give to our blood to get rid of it. But if it's at rest, these are the values. And so that is the values of the blood when it leaves. And then the process continues. These numbers you need to know. You need to know the partial pressures of oxygen and CO2 in all of these environments. All right. Questions on that? All right, excellent. Here was that picture I was talking about. Notice here, when the blood first enters the pulmonary capillary to the time it leaves, as I mentioned, is less than, oops, let me erase that, is less than a second. But notice, it only takes about a quarter of a second for that gas to diffuse, that oxygen to diffuse into the blood, and the partial pressure of oxygen to increase dramatically, all the way up to its max. Why is this significant? Why is that rapid diffusion of oxygen into the blood happening so quickly? Why is that important?
Normal functioning of the body.
True, that is important why we need to get the oxygen in there, but why do we need to get it in there so quick?
Get rid of CO2.
No, it's not related to CO2. Remember, CO2 and oxygen don't really affect each other that way. All right, let's take it a step further. Notice it takes this little less, 3/4 of a second to get through the pulmonary capillary while you're sitting here in the class, hopefully sitting. Some of you may be walking on a treadmill or something. Some of you may be at a standing desk. But for all of you that are sitting, this is about the time it takes your blood to get through your pulmonary capillary. Because what's your heart rate right now?
Around 76.
76. Let's make it 80 because it's a nice simple round number. 80. Let's say it's 80. Your resting heart rate is 80. And let's say instead, because I'm talking for 4 hours, you decided to get on the treadmill and run on the treadmill while you were listening to this lecture. What would happen to your heart rate?
Goes up.
Yeah, what if your heart rate doubled to 160? Heck, what if your heart rate tripled to 240? Instead of running on the treadmill, maybe you're running from a bear with an axe. If your heart rate tripled so that your heart was beating at 240 beats a second, would we still be able to fill up the oxygen into your blood if your blood was going 3 times faster through your body?
It should.
Yeah. Because it fills up with oxygen so quickly, we could essentially triple your heart rate, and you're still going to be able to fully perfuse your your blood with oxygen. Now, hopefully you're not doing too many things that are tripling your heart rate. But if you chose to do such a thing, right, there would be many problems that you would probably have as a result of that, but getting oxygen into your blood wouldn't be one of those problems. This process is incredibly efficient. All right. And again, remember it's 104 at the end of the capillary, but then by the time it gets back to the heart, it's 100 because that pesky bronchial veins. All right, questions on that? All right. What do we want to do? Let's go ahead and do this. All right. Just too early to take another break, but we have some more important concepts to cover. All right. Now, this ability to get these gases to move kind of goes back to one of the questions that got asked earlier. Does every single person have exactly 40 millimeters of mercury of carbon dioxide in their blood? It is going to differ slightly, but let's even take it beyond that. Within you, does every single alveolus in your body, right? Remember, there are a million alveoli between your two lungs. Does every single one of them have exactly 104 millimeters of mercury of oxygen inside of it and exactly 40 millimeters of carbon dioxide inside of it?
No.
No, absolutely not. So that means that since there are going to be variations, There are going to be variations of the level of oxygen and carbon dioxide in each alveolus. And again, as we've talked about, we are relying, that's too big. We are relying on diffusion for our gas exchange to take place. We're not using any ATP. So that means each alveolus will vary in how efficient it will be in gas exchange. And that could be an issue. So luckily, we're really, really smart. So currently, while you're sitting here in class with me, are you personally monitoring all 1 million of your alveoli to see what's going on and what the gas conditions are in each one of them to know whether or not it should be used for gas exchange? I hope not, because then you're not paying any attention to me. But since gas exchange is so vitally important and is a passive process, do we need to be monitoring the conditions inside of every single one of those 1 million alveolus? Yeah, absolutely we do. And luckily, we have a very important intrinsic process for monitoring and regulating gas exchange in every single alveolus. And this regulatory process is what we call ventilation, perfusion, coupling. Now, obviously, coupling means to bring together. So we need some quick definitions. We need a definition of ventilation, and we need a definition of perfusion. Anyone know what ventilation refers to?
Gas goes in and out.
Excellent. It is the rate of gas flow into and out of the alveolus. And what is perfusion? Excellent. The rate of blood flow into and out of the pulmonary capillary. Excellent. So ventilation is the rate of gas flow into and out of the alveolus. Perfusion is the rate of blood flow into and out of the pulmonary capillary. So we have our two definitions. Now we need our two regulatory processes. But really, are two regulatory rules. There are two rules, and these two rules are independent. Meaning one rule does not affect the other. Rule 1, the partial pressure of oxygen in the alveolus regulates perfusion, blood flow. Rule 2 is that the partial pressure of carbon dioxide regulates ventilation. airflow. Questions on that? All right. If there's no questions on this, then let's see how this works. And let's start first with rule 1. Partial pressure of oxygen regulates perfusion. What did perfusion have to do again with?
Blood flow.
Blood flow. Absolutely. Perfusion has to do with blood flow. And in this case, it regulates perfusion by changing the diameter of the arteriole or pulmonary, let's say it that way. Let's see how this might work. Here we have an alveolus. Oops, nope, don't want red. Here we have an alveolus. Here we have a pulmonary capillary. And let's say, for instance, in this alveolus, we had a high partial pressure of oxygen. If we had a high partial pressure of oxygen, would this gas be good for gas exchange or bad for gas exchange? Is this going to be good air or bad air? What do you think?
Good air.
Good air, absolutely. This is going to be good air. If this is good air for gas exchange, would we want more blood to come or would we want less blood to come to this particular capillary?
More to come to take more oxygen.
Right. We want more blood here. So if we have more blood, we get more gas exchange because we want that good gas exchange. And so we, of course, want more we want to increase perfusion. And how would we increase perfusion in this case? How would we get more blood to go into this capillary? Bingo. We are going to dilate. Oops. The arteriole. If we dilate the pulmonary arteriole, we are going to bring more blood to the area, more perfusion, more gas exchange is going to take place. And that's a good thing.
How you do that? How?
Again,
How to make it dilate?
if it's an intrinsic system. So remember, all of these changes are occurring here. Basically, there are chemoreceptors here in the alveolus that are measuring the oxygen levels. And as based on the oxygen levels, they release nitric oxide. More oxygen in the blood vessel, they release more nitric oxide that dilates the arteriole, and we get more blood coming to the area.
Okay.
Again, this is intrinsic. This is happening local. That's why you don't have to think about it consciously. You're not even thinking about it subconsciously. Every single alveolus is monitoring the condition of their own blood and monitoring airflow and blood flow at a local level. This is an intrinsic process. That's how every single alveolus is able to do this independently. So at any one time, more blood is going to be going to some alveoli than others. Let's look at the flip side of this. We have an alveolus. That's horrible. That's horrible too. Apparently I can't draw anymore. I didn't give myself enough room anyway, so I guess it doesn't really matter. I'm gonna have to redraw this anyway. All right. Now, let's say, for instance, we have a low partial pressure of oxygen inside of this alveolus. Is this going to be good air or is this going to be bad air for gas exchange?
Bad.
It's going to be bad air for gas exchange. So are we going to want more blood or less blood come into this capillary?
Less.
Want less blood come into this capillary. So we are going to decrease perfusion. And guess how we're going to do that? How would we decrease perfusion in this capillary?
Constrict in the.
There you go. It is going to lead to the constriction going to constrict the arteriole. And when it constricts the arteriole, less blood is going to come to the area, and we're going to decrease perfusion. What we're saying to the blood is this one here is not good for gas exchange. Go somewhere else. So notice the partial pressure of oxygen inside the alveolus regulates perfusion, regulates blood flow. By either dilating or constricting the blood vessels, we can either get more blood to come to this one for gas exchange or less blood to come here for gas exchange. And again, remember I mentioned this is an independent rule. Notice what we did not talk about at all here is carbon dioxide. Because does carbon dioxide play any role in whether we dilate or constrict the arteriole, whether we change the blood flow to this capillary? It plays no role at all. Oxygen and oxygen alone affects blood flow. All right. Questions on that one? I always worry when everybody's deadly quiet, especially on things that I know that sometimes people have challenges about. No questions. You all totally understand this concept. I know I'm doing a truly amazing job of explaining it, so that maybe that's just what it is. All right, let's move on to rule 2 then. Rule 2 involves the partial pressure of carbon dioxide. So again, let's go ahead and keep our alveoli, keep our blood vessels. I'm going to cheat a little bit. I'm going to bring this down. I'm going to bring this down. Bring that down. Bring that down. Bring this down. So, oh, and bring that up. Bring, oh, not that one. I want that there. This is what I want to bring up. Excellent. Go ahead and get rid of that. All right, so we talked about rule 1. Let's talk about rule 2. Rule 2 is the partial pressure of CO2 regulates ventilation by changing the diameter of the terminal bronchiole. So let's think about this. Let's say, for instance, we have air inside of this alveolus that has a low partial pressure of CO2. From what we know, would this be good air or would this be bad air if it had a low partial pressure of CO2?
Good.
Yeah, this would be quote unquote good air. And if we have good air in this alveolus, do we want to lose the good air in this alveolus?
No.
No, we want to keep the air in there. So if we want to keep the air in there, do we want to increase or decrease our ventilation?
Decrease.
Decrease ventilation. And guess how we would decrease ventilation in this case to stop this air from leaving? Excellent. Bronchoconstriction. I love that. Absolutely. We would constrict the terminal bronchiole. We would constrict the terminal bronchiole and ventilation would decrease. Conversely, if we have if we have air that has a high partial pressure of CO2, would that be good air or bad air?
Bad.
This would be our quote unquote bad air. And if we have bad air in here in this alveolus, do we want to keep this bad air in the alveolus?
No.
No, we want to get rid of it. So in this case, we want to increase the ventilation. And how would we increase ventilation in this case?
Dilate the terminal bronchiole.
Dilate the terminal bronchiole. Bingo. So there you go. Those are our two independent rules. Oxygen regulates perfusion by changing the diameter of the pulmonary artery, and carbon dioxide regulates ventilation by changing the diameter of the terminal bronchiole. Now notice, our bad air should be both low in oxygen and high in carbon dioxide. And in that case, we would want to constrict the blood going to it and then also dilate it to get fresh air in. And as soon as that fresh, good air came in, it should have a high level of oxygen, which would dilate the blood vessel, bringing more blood to the area. And we'd want to keep that air, so we'd constrict the arteriole. But so again, they should be related, but they don't have to be. They can be independent. If we had an alveolus that had high oxygen and high carbon dioxide, we would have an increased blood flow there, and we would have an increase in the diameter of the terminal bronchiole to get that CO2 out as well. So again, they are obviously related, but they're not reliant on each other, if that makes sense. I've done this with all my words and all the pretty pictures for this, but let's look at the pretty pictures from your textbook. So again, ventilation involves the amount of gas, the movement of gas reaching the alveolus. Perfusion involves the blood flow reaching the pulmonary capillary. Because gas exchange is passive and does not use ATP, we have to tightly regulate it at the local level using these intrinsic auto-regulatory processes. All right, the two rules. Change in the partial pressure of oxygen in the alveolus changes the diameter of the terminal arteriole. Changes in the partial pressure of CO2 in the alveolus changes the diameter of the terminal bronchioles. I don't know why I have terminal there, but not there. Let's fix that. And again, we can see how this works. Notice this one's got some bad air in it, but it's got a large blood supply to it, and that's a bad thing. So our oxygen levels are low. We need to constrict that arteriole and decrease blood flow to it. Then we need to dilate the alveolus. And when we dilate the alveolus, we get more oxygen in there, better air in here, that good air. And then once again, we have to dilate our arterioles and get the blood flow in here so that we're constantly able to match. We're constantly matching ventilation and perfusion, getting rid of bad air, bringing in good. bringing blood to the good areas, getting rid of blood in the bad areas. And it's dynamic and it's local. So this is happening in all 1 million of your alveoli constantly. All right. And again, got the pretty pictures from a textbook that show all of that. All right. Questions on that? All right. Here's the good news. We've gotten through a lot of physiology so far. Here's the bad news. It's going to get worse. So now what we need to do is talk about how we know how to get oxygen into and out of the blood. We know how to get carbon dioxide into and out of the blood. But as we've hinted at a couple times, and as you were learning when you were doing your lab exercises at home, there's some special things that happen to oxygen and carbon dioxide when they enter into the water, into the plasma of our blood. And so we need to talk about the mechanisms that allow them to be transported. Now, before we get to the really challenging stuff of this section, we need, I think, another break. If I can get my phone to unlock. Apparently it suddenly doesn't like my finger anymore. Excellent. Looks like it's, well, let's call it 2:15. So we'll come back at 2:30, and at 2:30, we will pick up the lecture from here. So we'll restart at 2:30, and I will start the recording at that point. All right. Any questions on anything we've covered so far, ventilation, perfusion, anything before we take our next break? All right. I will see you guys in 15 minutes. All righty. So let's go ahead and get started. We were talking about transportation of oxygen and carbon dioxide. And let's start with oxygen first, because oxygen is much easier to understand. Basically, 98.5% of all the oxygen that is transported by your blood is transported by hemoglobin. Right? Remember, we talked about how our hemoglobin has 4 subunits that are the same size. At the center of each one of them is a heme group. And the center of each of those heme groups is an iron. Every single one of these irons at the center of our hemoglobin is capable of binding 1 oxygen. So a single hemoglobin can bind 4 oxygens to it. And anybody remember about how many hemoglobins are in a single red blood cell? Microphone's on, right? Yeah, much more than that. Remember, close to 250 million hemoglobin inside of a single red blood cell. So that's like 1 billion oxygen that can be carried just by 1 single red blood cell. And if you think about how many red blood cells are in a single drop of blood, you can see the massive oxygen carrying capacity of our blood. However, the good and the bad of that is that all of that oxygen, almost all of it, is bound to the hemoglobin. Right? When, of course, it does that, we call it oxyhemoglobin, HbO2. And that doesn't leave a lot left. Right, so the remaining, I guess I didn't write that here, so we'll do it here. The remaining 1.5% is just dissolved in the blood, in the plasma, in the water, right? Because that's basically what plasma primarily is. And there you go. Oxygen is just that simple. Now, CO2 is a little bit more complicated. There is a small amount dissolved in the plasma, just like in oxygen, although it's closer to 7 to 10%. And some of it is also bound to the hemoglobin. I'll just redraw one of our subunits. Here's just one of our subunits. With that iron in the center. Now, remember, the iron is where the oxygen binds. Where a CO2 molecule will bind is on the heme as well, but in a different location, right? It's not like musical chairs where they're fighting for the same seat. It's more like the couch downstairs with my two daughters, right? Is it possible for both of my daughters to sit on the couch at the same time? Absolutely. There's plenty of room on the couch for both of them to be on the couch at the same time. However, if one of them is already on the couch, does the other one like to sit on the couch when the other one's already there? No, right? So her being on the couch, Big being on the couch deters Little from being on the couch. When Little's on the couch, Big doesn't want to sit there. It's capable of both of them being on there at the same time. But typically, if one's on there, it doesn't stop the other, but it discourages the other. And that's what happens here. Our oxygen will bind out here on the iron. And after all, that's what makes the red blood cell red. And the CO2 binds out here on the side of the hemoglobin. So they're not fighting for the same seat, but when one binds, they make it a little harder for the other one. So when we have a carbon dioxide bound to the hemoglobin, we call it carbaminohemoglobin, which you got to figure just looking at the number of letters in that is something you're eventually going to have to spell at one point or another. But on an essay question, you could then abbreviate it HbCO2. But notice even that is only about 20%. However, as you know, because it was a big part of one of the two activities you did for today's lab, you know that there is this magical equation where when carbon dioxide is added to water, it forms an acid. And so what ends up happening is about 70% of the carbon dioxide is converted into this acid. And again, it's not just any acid, it is carbonic acid. So carbon dioxide combines with water to form carbonic acid. Carbonic acid, like all acids, disassociates. And when it disassociates, it forms hydrogen ions and a special molecule, HCO3 negative, which is bicarbonate. And so again, one of those fancy equations that you should have been using and talking about in the lab you were talking about. Yeah, well, no. So again, HCO3 is bicarbonate. Carbonic acid is H2CO3. Bicarbonate is HCO3 negative.
So bicarbonate is the primary transport, right, for CO2? Because
no. What happens is, yes, I see what you're saying. Oh, yes. So what happens is, if you think about it, so let's think about it in terms of the internal gas exchange. The key to this reaction is it is a reversible reaction. So let's think of it in terms of at the tissues. And let's make this blue to emphasize this. When our blood gets to the tissues, as we know in that systemic capillary, there is going to be an increase in CO2 that enters into the blood. Right? We know that's what happens at the systemic capillary. So basically what happens is we get an increase in carbon dioxide in the blood. And when we get that increase in carbon dioxide in the blood, it drives the chemical reaction to the right. And so what happens is our CO2 is converted into hydrogen ions and bicarbonate ions. So basically our carbon dioxide is converted into hydrogen ions and bicarbonate ions. Acids don't like to stay together in water. They like to disassociate. So when the tissue is in that systemic capillary, when the blood is in that systemic capillary at the tissues, it's taking on carbon dioxide and it's driving the chemical reaction to the right. Conversely, when we get to the lungs, in a pulmonary capillary, when we're in that pulmonary capillary, there you're going to see a decrease in carbon dioxide in the blood because the carbon dioxide is being given out. And when that occurs, as a result of that, it's going to drive the chemical reaction to the left. So basically what's going to happen is, move this out of the way, grab this. As carbon dioxide leaves into the lungs across into the air, then our chemical reaction is driven this way. Bicarbonate is converted back into carbonic acid, which disassociates in carbon dioxide, and that carbon dioxide diffuses across the respiratory membrane into the lungs to be given off. So what happens is we have this reversible reaction, and the direction this reversible reaction is going to go is going to be primarily based on the carbon dioxide in the area. So when carbon dioxide enters the blood, it makes more bicarbonate ions, and then those bicarbonate ions are converted back into carbon dioxide to get rid of the carbon dioxide in the lungs. All right, questions on that? Now, like all chemical reactions that we are aware of, there is an enzyme that facilitates this. And in this case, that enzyme is carbonic anhydrase. And what's awesome about this enzyme is two things. One, it can actually facilitate the chemical equation in both directions. It works in both directions. So it can help to convert bicarbonate into carbon dioxide, and it can also convert carbon dioxide into bicarbonate. So it's really awesome in that it works in both directions. But the other cool thing about it is where it's found is in the red blood cells. Inside, let's say, be more specific, inside the red blood cells. After all, red blood cells are carrying all the oxygen. That's where the gases are going. So it makes sense that in those red blood cells that are going to the tissues and going to the lungs is a perfect location to have this enzyme. All right, questions on that? Now, as we know, when we have more carbon dioxide in the blood, what happens to the pH of the blood?
Drops.
So yes, absolutely. So and I want to go back to Alex's question, Jim. Yes, carbonic anhydrase both helps to, well, no, where carbonic anhydrase primarily functions is right here. Carbonic anhydrase helps to convert carbon dioxide and water into carbonic acid, or carbonic acid back into carbon dioxide and water. Carbonic acid is an acid, and acids in water disassociate. Like salt. When you put salt in water, the sodium and the chloride separate from each other, right? It doesn't require an enzyme or anything for that to happen. They just pull apart, right? They separate in the water, and that's what acids do. Acids are hydrogen donators. So in water, they release their hydrogen ion. So the carbonic anhydrase really helps with this chemical reaction of taking that carbonic acid and turning it back into carbon dioxide and water, or taking carbon dioxide and water and converting it into carbonic acid. All right. And yes, you guys are correct. When there's more CO2 in the blood gets more acidic. And of course, that means that the pH decreases. And then when there is less CO2 in the blood, it's more basic, more alkaline, and our pH increases. Excellent. All right. Questions on that? All right, excellent. So if you think about it, and again, I'll show you the picture again, but let's cheat and actually go all the way back to it. Oops, too far. There we go. Notice here we see that process. Bicarbonate ions are converted into carbonic acid, which becomes CO2 and water, and that CO2 is released. Or when CO2 enters the blood, it mixes with the water, becomes carbonic acid, and disassociates into hydrogen and bicarbonate ions. So we'd seen this before, and we have an even nicer, prettier picture of this in your textbook that we'll look at in just a second. Did all of that. Excellent. All right. The last thing we need to talk about for this transportation, though, is a big one. It is our oxygen hemoglobin dissociation curve. Now, as we mentioned, in a single hemoglobin can bind how many oxygen?
4.
Excellent. It can bind 4 oxygen. So if we think about this, and let's actually go to the whiteboard for this. Let's do some drawing. So no, I don't want it to be green. Let's move this over here. Change this to black. All right. What we are drawing right here is the oxygen hemoglobin disassociation curve. Now, as we just mentioned, a single hemoglobin could bind as many as 4 oxygen. So what we're going to put over here on the y-axis is the percentage of oxygen bound to the hemoglobin. All right. Now, the good news is this should be pretty easy. If there were 0 oxygens bound to the hemoglobin, what would our percentage bound be?
0.
0%. There you go. Excellent. Oops. If one oxygen was bound to a hemoglobin, what would our percentage be then?
25.
25. Excellent. If 2,
50.
yeah, I was gonna say, these questions should be getting easier. If 3,
75.
and if 4 were bound,
100.
excellent. So notice these are good, easy landmarks. 100%, 75%, 50%, 25%, and 0%. And again, that would be the number of oxygens that would be bound. So let's move this here, make it all nice and pretty. And then we could remind ourselves that this point would be 4 bound. This point would be 3 bound. This point would be 2 bound. This point would be 1 bound. And this point would be none bound. Excellent. Now, this is, of course, when we are talking about one hemoglobin. Is it easy to, as we're measuring this as human beings in the real world, what's going on in one single hemoglobin? No, of course not. So when I'm measuring the blood and I say that the oxygen hemoglobin dissociation is at 75% bound, even if I'm just talking in one red blood cell, does that mean that every single one of those 250 million hemoglobin all have precisely 3 in them? No, of course not, right? But that means on average they are. There may be 1 or 2 that have 4, there may be 1 or 2 that have 2, but by the vast majority, and certainly by average, all of them are going to have basically 75. It's obviously not going to be perfect. And think of it this way. If I have a certain percentage where half of them have 3 bound and half of them have 2 bound, what would my percentage be there?
Like 62.
Yeah, about 62 and a half, something like that. So it would be somewhere like within here, right? Like halfway in between here. At this point on the curve, at that point, half of them would have 2 and half of them would have 3. Does that make sense? So basically, we could be anywhere on this curve based on how many on average are bound to all of the hemoglobin total. So it isn't just 4 points. You can be anywhere in between on this curve when you're dealing with millions of hemoglobins. All right, does that make sense? Hopefully. All right. So now the big question becomes, we know that the first factor we care about is how many oxygen are bound to the hemoglobin. But the next question we have is, what is the primary factor that determines how much oxygen is bound to the hemoglobin? Anyone have an idea of what that primary factor might be?
If there's already a carbon dioxide bound to it.
Well, carbon dioxide can't change it, but carbon dioxide, remember, is not fighting for the same spot.
But they don't like being on the same one, right?
True, they don't like. So can they affect each other? Absolutely, right? But affecting is not the same as the primary factor. Alexa's got it, but let's take it to an extreme. If you had a red blood cell sitting inside of a blood plasma, and that blood plasma had no oxygen in it. If the blood plasma had no oxygen in it, how much oxygen would be binding to the hemoglobin? Yeah, it's not a trick question. None. None would be binding to it. Absolutely. However, if we had a red blood cell in an environment that was just chock filled, had massive amounts of oxygen in that blood plasma, do you think that blood plasma, that oxygen in the blood plasma would be able to bind to the hemoglobin and you'd have a massive amount? Absolutely. So as was pointed out, the primary factor that determines it is the partial pressure of oxygen. The partial pressure of oxygen is the primary factor that determines how much oxygen is bound to the hemoglobin. The partial pressure of oxygen, right? This kind of makes sense. Basically, what it's saying is how much oxygen is surrounding the red blood cell, or the hemoglobin, I guess, in the plasma. As we already mentioned, in an extreme case that hopefully doesn't happen in your body, oops, don't want that to be purple, that would be 0. So the worst case scenario would be a partial pressure of oxygen that was 0. And as we already guessed, if the partial pressure was 0, what would the amount of oxygen bound be? 0. So that would be our starting point right there. 0 and 0. Excellent. Now, let's think about this. From a biological standpoint, what is the highest partial pressure of oxygen we would normally see inside the body? Are there ways to artificially inflate it? Yes. But in a normal, healthy, functioning body, what do you think the highest level, the highest partial pressure of oxygen we see in the body would be? 160 is what it is in the atmosphere out here, but is that what it is inside the lungs where the gas exchange takes place? 104. Excellent. Now, again, could we, by putting someone on an oxygen ventilator or something like that, increase that? Sure. But if you think about what the normal value in a normal biological situation would be, that would be 104 millimeters of mercury. And guess what the percent bound at 104 millimeters of mercury would be for the hemoglobin? How many oxygen do you think are binding to the hemoglobin when the partial pressure of oxygen is 104 millimeters of mercury? Yeah, well, it is indeed. It's essentially 100%. It's a little under 100%, but for you're right, it is 98. But for our argument's sake, we'll put it there at essentially 100. It's a tiny bit under 100, but essentially almost all of our hemoglobin are going to have 4 oxygen bound to it. All right, those are the two extremes, and hopefully those make some semblance of sense. Yes, no, maybe?
Yes.
Okay, excellent. Now, normally, once you have 2 points on a graph, what can you do?
Draw the line.
Draw a line between them, absolutely. Normally, when you have 2 points on a graph, you can draw a line on them. But remember, this is called the oxygen hemoglobin disassociation curve. Is that line a curve?
No.
No. And the reason is one more factor. As was mentioned earlier, carbon dioxide can make it harder for an oxygen to bind to the hemoglobin, but it turns out something helps it. When one oxygen binds, binds to a hemoglobin, it makes it easier for a second one to bind. And the second makes it easier for the third, and the third makes it easier for the fourth to bind. So as more oxygen bind to the hemoglobin, it makes it easier for other oxygen to bind. And the opposite is true as well. When one oxygen leaves the hemoglobin, it makes it easier for a second one to leave as well. And so because of that, we don't get a straight line for this. Instead, what we get is basically kind of a sigmoid curve that both I'm going to have trouble drawing because I'm a horrible artist, but also because it's a little tricky. But basically, we get this curve that looks nothing like that. Let's try it one more time. That's a little better, although not much. It's going to have to do. We'll look at the pretty picture from the textbook in just a minute. All right. So far, so good. This is our oxygen hemoglobin dissociation curve. In an extreme bad and artificial situation where there was no oxygen in the blood, of course, no oxygen could bind to our hemoglobin. That makes sense. And in a truly biological situation, when our hemoglobin is in that pulmonary capillary, in the lungs, when the partial pressure of oxygen is 104, it loads up with oxygen and binds 4 oxygens to the hemoglobin. Now, let's put another point on this map. What did we say the partial pressure of oxygen was going to be in a resting tissue? In that resting tissue, what was the partial pressure? Partial pressure of oxygen, we said, was equal to 40 millimeters of mercury. So I need to grab this. Nope, not you. You get to stay there. I need this one. Partial pressure of oxygen I'll put over here as a reminder. And that will allow me to put this one here. That looks about right. 40 millimeters of mercury. So let's think about what's happening here. What's happening here is our red blood cell, the hemoglobin, goes to the lungs. In the lungs, it loads up with 4 oxygen. Then it gets transported in the blood to a systemic capillary where the partial pressure of oxygen is 40. And at 40, what is the amount of oxygen that is bound to the hemoglobin? At 40 millimeters of mercury, what is the percentage? 75%. Excellent. So my hemoglobin goes to the lungs and loads up with 4 oxygen. Then it gets transported to our systemic capillary of tissue that is at rest. And what does it do when it gets to that tissue that is at rest?
It dumps oxygen in it.
How many oxygen?
40. Or no, wait.
How many oxygen is going to be bound to my hemoglobin at that in systemic capillary where the tissue's been at rest? 75%. How many is going to be bound? 3. So how many does it give off? 1. So it releases 1 oxygen, and our resting tissue gets 1 oxygen from this 1 hemoglobin. Then my hemoglobin gets transported back to the lungs. And when it gets transported back to the lungs, guess what it does? When it gets to the lungs and the partial pressure is 104, it gains an oxygen. And then it gets transported to another resting tissue in the body. And when it gets that resting tissue in the body, what does it do? These questions should be getting easier. Gives off an oxygen to that resting tissue. And then it goes back to the lungs. And what does it do when it goes back to the lungs? Picks up an oxygen. And then it gets transported to yet another resting tissue. And when it gets to that resting tissue, what does it do? Gives up an oxygen. And then it goes back to the lungs and picks up an oxygen. And then something interesting happens. It gets transported to a location of your body that is vigorously working, right? First, it went to your right leg, which isn't doing anything. Then it went to your left leg, which isn't doing anything. Then it went to your left arm, which isn't doing anything. But then it went to your right arm, where you're furiously taking notes. Or maybe to your brain, where you're furiously firing action potentials. And when we have active tissue, that active tissue could have a partial pressure of oxygen that is low enough that it's maybe 17 millimeters of mercury. And so when it gets to that active tissue where it's 17 millimeters of mercury, notice what happens there. That hemoglobin gets to that active tissue coming from the lungs where it had 4 oxygen. And what's going to happen to it when it gets to that systemic capillary where the tissue has been active? Come on, baby birds, give it to me. Excellent. It's going to give off 3 oxygen. Notice the tissue that was at rest, it only gave 1 oxygen to. But this tissue that it's active, it's giving 3 oxygen to. The beauty of this system is that what happens here is each tissue gets oxygen based on its need. And of course, what determines its need? The partial pressure of oxygen. If the partial pressure of oxygen is low, it gives it more oxygen. If the partial pressure oxygen is high, it gives it less oxygen. And if the partial pressure is really high, it loads up with oxygen. So the beauty of this system is that every single tissue in your body is going to get the amount of oxygen that it needs based on how much oxygen it already has. If it has a moderate amount of oxygen, it only gets a little. If its oxygen levels are low, it's going to get a lot. I need to make sure this makes sense to you because this is about to get a whole lot worse. So if this doesn't make sense, you need to say something now. Any questions on this at all? Absolutely. What this curve shows us is how much oxygen will bind to a hemoglobin. And it shows us that the primary factor that determines how much oxygen binds to a hemoglobin is how much oxygen is available in the blood plasma. And that amount of oxygen that is found in the blood plasma is the partial pressure of oxygen. So if we look at our 2 extreme situations, if there was absolutely no oxygen in our blood plasma, well, then that makes total sense. There'd be no oxygen to bind to our hemoglobin, and so the percentage would be 0. In an environment like the pulmonary capillary, where the partial pressure of oxygen is very high, 104 millimeters of mercury, there's a ton of oxygen available. And so not surprisingly, our hemoglobin fills up completely. Again, someone mentioned it's actually 98%, but for all intents and purposes, they all load up completely, 100% with 4 oxygens. All right. Now, once it's in the lungs, it gets loaded up with that oxygen. It's now that oxygen-rich blood where my hemoglobin has those 4 oxygen bound to it. But as we know, this hemoglobin is now going to be transported someplace in your body. And that place in your body is going to have a different amount of oxygen in it. If, for instance, like right now, while you're sitting in the chair, this hemoglobin is transported to your legs, right? Unless you've had too much caffeine and you've got that Jimmy leg going on, your leg's sitting there at rest and not doing a whole heck of a lot. Because it's not doing a whole heck of a lot, as we know, the partial pressure of oxygen in that environment is 40 millimeters of mercury. And when the environment is 40 millimeters of mercury, then only 3 oxygen can bind to the hemoglobin. Well, the hemoglobin's got 4. So what does the hemoglobin have to do? It has to release 1. So it releases that 1 oxygen, and that 1 oxygen is given to the tissue. It then goes back to the lungs. When it goes back to the lungs, my hemoglobin can bind 4 oxygen, but it only has 3 on it. So basically it picks up a 4th oxygen. But remember, how much oxygen is bound to the hemoglobin is dependent on the environment. If instead this hemoglobin is next transported to your right arm, where you're furiously taking notes, that tissue is very active. And because it's very active, the oxygen levels are lower. Partial pressure of 17 millimeters of mercury. In that environment, only 1 oxygen can bind to the hemoglobin. So that means the hemoglobin is going to let go of 3 oxygens. And those 3 oxygens are going to be given to the tissue. And our hemoglobin will only have 1 left. It will be at a 25% bound. Of course, when it gets back to the lungs, it can bind 4. It only has 1. So it's going to pick up 3 more. And so in this fashion, how much oxygen we give to the tissues in the systemic capillaries are totally dependent on what the partial pressure of oxygen is in that environment. And the beauty of this then is that every single tissue, how much oxygen your right hand is getting versus your left hand is getting, how much oxygen your right hand is getting versus your right foot is getting, is going to be different based on how active that tissue is. The more active the tissue is, the lower the partial pressure of oxygen, and the more oxygen it'll get from the hemoglobin. All right. Questions on that? All right. So, as we know, the more active the tissue, the less oxygen that binds to the hemoglobin. All right, excellent. So that is our big rule. The more active the tissue, the less oxygen that binds to the tissue in that environment. And obviously, as we said, the primary factor that tells us that the tissue is active is the partial pressure of oxygen. If there is, we'll do it over here. If the tissue is more active, then the partial pressure of oxygen is lower. All right, that's the main factor. Whereas if the tissue is less active, then the partial pressure of oxygen is higher. All right, so far so good with that.
Partial pressure oxygen in blood, right?
Yes, well in the plasma in that environment, yes. Absolutely. However, in a tissue, in that interstitial fluid, is the partial pressure of oxygen the only indicator of tissue being metabolically active? Well, I'm asking the question, so what's the obvious answer? Are there other factors that can indicate that a tissue is active?
Yes, should be.
Yes, absolutely. The problem, so while this is our starting point, there are other factors that can indicate the tissue is active. And since there are other factors that can indicate if our tissue is active, do we want to rely just on the oxygen level to determine how much oxygen we give to our tissue?
No.
No. What is another factor that could indicate that a tissue is active?
Build up of CO2.
Excellent, perfect. Okay, so stop. That's 3 really good ones right there. So let's start with those, or 2 really good ones there. CO2 levels. If the tissue is more active, would CO2 levels go up or CO2 levels go down?
Up.
So the partial pressure of CO2 would increase. Excellent. And if the tissue is less active, then the partial pressure of CO2 would decrease. Excellent. All right. I saw pH, the acidity. If the tissue was more active, would pH increase or would pH decrease?
Decrease.
Decrease. It would get more acidic. Right? And so if the tissue is less active, our pH would increase. Right? Less acidic. What would another indicator of activity of tissue be? Temperature, absolutely. So what would happen to temperature in the tissue if it was more active?
Go up.
Increases. And so, of course, if the temperature goes down, then that's an indication that tissue is less active. Any other factor? This last one may be a little trickier to come up with.
What about the glucose breakdown?
Okay, you're getting close, absolutely. But remember, glucose is going to be inside of the cell. So would it be easy in the interstitial fluid to see how much glucose was available?
No.
No, but you do have the right idea. When tissues are active, aren't there metabolic wastes?
Increase.
And what would happen to the number of metabolic wastes if our tissue is active?
Increase.
Increases. Excellent. Whereas here, if the tissue is less active, the number of metabolic wastes would decrease. So notice there are 4 other factors that could show us that the tissue is more active or less active. And notice how cool this is. If it's less active, CO2 decreases, temperature decreases, metabolic waste decreases. Yes, pH goes up, but remember the pH going up tells us that we have a decrease in acidity. When the tissue is more active, we have more CO2, more temperature, more metabolic waste, and more acid. So that part makes it easy. So the question is, how do these things affect the curve? Well, before we figure out about how it affects the curve, let's think about what we want the effect to be. If the tissue is more active, do we want to give it more oxygen or less oxygen?
More.
We want to give it more oxygen. If we want to give it more oxygen, do we want more bound to the hemoglobin or less bound to the hemoglobin?
Less.
Excellent. And so that's what happens when one of these other factors, the CO2, the pH, the temperature, or the metabolic wastes increase. Then what happens to our curve is that our curve shifts down. So in this case, our curve is going to shift down. And when it shifts down, we release more oxygen at every point of the curve. So basically what happens is this shifts down, and 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 this shifts down. All of our points shift down. At every single point along this, it shifts down. And notice if we connect all those points, while technically our points have moved down, if you look at it, it looks like our curve shifted to, to the right. So it looks like the curve is shifted to the right, but really what has happened is it is curved down. And so you can say that the curve has come down or the curve is shifted to the right. Both of those would be acceptable. Conversely, if our tissue is less active, then the effect we want is we want to give less oxygen. If the tissue is not being active, we don't need to give it as much oxygen. And so we want more bound to the hemoglobin. And if we want more bound to the hemoglobin, which way do we shift the curve?
Up.
Curve up. Absolutely. So every point along the way, we are going to shift the curve up. Because we want to give it less oxygen. But again, when you actually take the time to connect all those dots, it looks like the curve shifted to the left. All right. We've done this on the board here. And again, I apologize. I've done the best I could do with my drawing skills. But let's take a look at the pretty pictures of this from your textbook. As we know, 4 oxygen can bind to a hemoglobin. It's a curve because every time one binds, it makes it easier for the other to bind. And it gives us a percent bound, which we call our hemoglobin saturation. And the primary factor that determines how much oxygen binds to a hemoglobin is the partial pressure of oxygen. And there is the pretty curve. Notice at 100, 104, it's essentially 100, like I said, 98 just under. But for all intents and purposes, we load up with 4. Notice at 40, when the tissue is at rest, partial pressure of 40, it's at about 75%. But at a partial pressure of 17, it's at 25%.
May I ask you?
Yes.
Shouldn't it be like the need of the tissue
in the oxygen, not in the pressure of the oxygen in the blood.
Well, but see, remember, at the capillary, whatever the partial pressure of oxygen in the interstitial fluid is, it's going to drive the partial pressure of oxygen in there to be what it is.
Oh, yes, yes, I remember. Okay, thank you.
So that's what, so exactly, that's why it changes in that environment. We'll talk about BPG in just a second. Great question, we will get there. So again, here we see those two examples, the tissue at rest, the tissue exercising, and the tissue that is exercising that is more active, is going to get more oxygen. So this is our oxygen hemoglobin dissociation curve, period. But as we talked about, there are other factors that can modify the curve. So when we're describing the curve, you talk about what's on the x-axis, you talk about what's on the y-axis. These are the things that affect the curve. However, then as we can see, we can modify the curve. The first factor that we talked about that can modify the curve is the partial pressure of carbon dioxide and pH. Now, obviously, are these two things related to each other? Yes, the more CO2 you have, the more acid you're going to have. But remember, we also talked about there's lactic acid and other things as well. So these are different factors. And the effect the pH has on the blood is actually called the Bohr's effect. I don't care if you actually know that or not. But here's our normal curve. Notice when hydrogen ions go up, when it becomes more acidic, when CO2 goes up, our curve is shifting. And again, remember, this shift is technically down. Every single one of these points, the curve is shifting down at every single one of these points. But when you put them all together, it looks like the curve shifts to the right. Conversely, when there's less carbon dioxide or the blood becomes more basic, then every single point on the curve goes up. But notice when you draw the line, putting them all together, it looks like the curve has shifted to the left. Same thing is true for temperature. Notice the warmer the temperature goes down, colder temperature goes up. Notice this is part of the problem with frostbite. Remember, we talked about frostbite way back in 430 when we were talking about the integumentary system. We talked about how the blood vessels in your extremities will constrict. And as they constrict, less blood is going to that tissue. But notice we said not no blood, just less blood. Less warm blood, so the tissue gets colder. But notice also the blood that's going there, if the blood is really, really cold, when it gets to the tip of your finger, is that blood going to want to give off its oxygen? Can it give off its oxygen when it gets to that environment?
No.
No. And if it can't give off its oxygen, not only is the tissue really cold, but it's not getting the oxygen that it needs, and that tissue dies, right? You get that horrible frostbite on the tip of your nose or your ears, your fingers, or your toes, because they're not getting the warmth and they're not getting the blood that they need. Well, they're getting the blood, they're not getting the oxygen that they need. And what was the fourth factor again? pH, carbon dioxide, temperature, and
metabolic waste.
Metabolic waste, absolutely, metabolic waste. One of the common metabolic wastes, and this answers your question, Nabil, is biphosphoglycerate. Biphosphoglycerate is actually a metabolic waste produced by, ironically, red blood cells. So when the tissue is more active, it has more of this biphosphoglycerate present. And this, so it's an example. So BPG is just an example of one of the metabolic wastes that can have this type of an effect as well. It's a common one because it's associated with red blood cells. But other metabolic wastes can have a similar effect. More metabolic waste, shift to the right, give more oxygen. Less metabolic waste means we're less active, give less oxygen. And there you go. So again, formed in red blood cells during glycolysis, an indicator of activity. So it's a metabolic waste. All righty. Questions on that? All righty. Excellent. So just that easily, we have talked about our oxygen hemoglobin disassociation curve. Now, obviously, on an exam here online, you're not going to be able to draw this. If we were doing this in the classroom, I would guarantee you're going to be drawing this. Obviously, I'm not going to make you draw this because I'm not going to make you use the whiteboard for that. But are you going to need to be able to describe these? Do you think this is going to be an important essay question that is very, very likely in some fashion or another to show up on your exam? Yeah, I think so, probably too. All right, excellent. Oh, great question. How does, we talked about carbon monoxide poisoning, and how do you think carbon monoxide affects this curve? What do you think carbon monoxide would do to this curve?
You wouldn't be able to refill your hemoglobins in the pulmonary capillaries.
Yep. So basically, your curve would look like that. Absolutely. Doesn't matter how much oxygen is in the blood, is in the plasma, you're not able to bind it to the hemoglobin. And that's the problem. Because remember, as we talked about, 98.5% of the oxygen that is being transported by your blood is being transported on your hemoglobin. So the 1.5% of oxygen that is just dissolved in your blood plasma, is that going to be enough to give the tissues the oxygen that they need to do their job?
No.
No, absolutely not. And so that's why that carbon monoxide poisoning is so incredibly dangerous, because it blocks the oxygen from binding to the hemoglobin and basically destroys your curve. Excellent. All righty. Oh, here are the pretty pictures that I promised you. Notice, again, this is what is taking place in our internal gas exchange, right? This is where our oxygen is being released from the hemoglobin and dissolving out to the tissues. The tiny bit that's in the blood plasma is leaving as well. And our blood is loading up with oxygen. Loading up with CO2. Some CO2 is binding to the hemoglobin, forming the carboamino hemoglobin. And remember, that can help to encourage the oxygen to let go. Some of it, remember, enters into the red blood cell, not to bind, but because in the red blood cell is where that carbonic anhydrase is, where it converts that CO2 and water into carbonic acid, which then disassociates and releases that bicarbonate into the plasma. Some CO2 is just converted naturally without the enzyme into carbonic acid and bicarbonate, but not nearly as easily without that enzyme. And remember, we said about 10% is dissolved in the plasma. Conversely, when we get to the lungs, the carbon dioxide from the plasma leaves to the lungs. Without the enzyme, it can be converted back into that carbonic acid can be converted back into carbon dioxide and released. But again, in the red blood cell with that enzyme, this conversion of carbonic acid into CO2 and water is much, much faster. And the ones that are on the red blood cell, on the hemoglobin, can be released, making it easier for that oxygen to bind. And a teeny bit of oxygen is dissolved in the blood. So notice these pictures from your textbook do an amazing job of not just showing us the internal and external gas exchange, but what's happening to the oxygen and the carbon dioxide, and how so that they can be transported from one location to another. All right, questions on that? All right, looking at the time, here's what's going to happen. We still have to talk about control of respiration. This isn't going to take too much time, but I appreciate that that curve and that stuff can be a little dense. So I think we'll go ahead and take one more quick break. We'll call it a 12-minute break. So we'll come back at 3:45. And then also, because this ran a little late, I'm not going to start the urinary stuff. We'll have plenty of time in the 3 days that we need to be able to cover it. So we'll just take a quick break. We will restart at 3:45. And at that point, we will finish off the rest of the lecture by talking about how we control respiration. So we'll start the recording at that point. Get a quick drink, get something quick to eat. Come back here in 11 minutes, 12 minutes, and we will finish off the lecture at that point. Yeah, I think I do. So yeah, that's a great idea. Let's stop at that point and leave it there. So let's switch to the whiteboard view. Excellent. And there you go. So then I'll just write this here. So we will restart at 3:45. Excellent. Any other questions? You're welcome, Dalton. All right. Any other questions? All right. See you guys in 11 minutes. All righty, home stretch. So you've learned many things that you wanted to know and many more that you didn't about the respiratory system. The last thing we need to do is talk about how we are going to control our rate of respiration. Our respiratory centers found here in our brainstem, as we can see, are what are responsible for controlling both how quickly we breathe, but also the depth, the rate and the depth at which we breathe. There are basically three main respiratory centers. There are two of them located within the medulla oblongata. One is known as the dorsal respiratory center or group, and the other is the ventral respiratory group. And again, these are called groups because these are clusters of nuclei. Remember, a nucleus is a cluster of cell bodies located, typically surrounded by white matter in our central nervous system. So these are a bunch of cell bodies where, of course, we know we process information. The other main location is in the pons, known as the pons respiratory center. So we have these three main regions that are responsible for producing what we think of as our normal resting respiratory rate. Now, each of them have slightly different functions. The dorsal respiratory group in the medulla is responsible for receiving massive amounts of inputs from our environment, right? Now, what would be some of the types of sensory information we would bring in? What do you think are the factors that need to be monitored or regulated? I've done in my chat window. Okay, heart rate could be one of the factors that can influence it. Although the heart rate is that necessarily going to directly affect what our respiration rate is, or is some factor that would affect heart rate also affect respiration rate? When we exercise, our heart beats faster and we breathe faster, but we don't breathe faster because our heart's beating faster. Both things are occurring because, excellent. So things like the partial pressure of oxygen, things like, so we need more oxygen, or it could be that we need to get more oxygen in, or what else could it be?
Excess of CO2.
Yeah. Get CO2 out. So the partial pressure of carbon dioxide. And of course, since we know carbon dioxide also influences the acidity, pH, things like those metabolic wastes we talked about. Excellent. All of those are the types of factors from going on inside of our body that could influence and modify the way that we are going to change our rate of breathing. Its job is then to collect this information and send it to our ventral respiratory group. And our ventral respiratory group is what produces our normal resting rate of breathing, right? What we call our eupnea. Eupnea is your normal resting rate of breathing, and it typically involves about a 2-second inhalation and a 3-second exhalation. All right, and a 3-second exhalation. So, and if you think about it, that takes 5 seconds. How many seconds in a minute? These are the easy questions, folks. Excellent. So 5 seconds of breath, 60 seconds per minute. What's your normal resting breath rate? How many breaths do you take a minute? Takes you 5 seconds to take a breath. There's 60 seconds in a minute. How many breaths do you take normally in a minute? 12. There you go. Excellent. 12. Some of you need to work on your math. 12. There you go. 12. All right? Normal resting breathing rate is about 12 breaths per second. Now, let's talk a little bit about how these inspiratory and expiratory centers work. And let's grab the whiteboard to do this because it's a very fun process. And by fun, I mean it's a neural circuit. Now, what if you're doing your normal resting breathing? What are the muscles that you want to control?
Your diaphragm.
Oops. Excellent, your diaphragm. And what was the other one that can help us with our normal resting breathing?
External intercostal.
Now, if our goal is to stimulate them, if we want to excite them, so let's make this a little bigger. So these are the muscles we want to control. When these muscles contract, what action occurs from a respiratory standpoint?
Inhalation.
And when they relax, then we exhale. All right. So these are the muscles we want to control. The diaphragm and the external intercostals. And so what happens is when we fire action potentials, those action potentials, and this I want to be smaller, we're going to fire those action potentials. They stimulate the muscles, as we know they're going to do, by releasing acetylcholine and generating that muscle action potential and everything else that goes along at that neuromuscular junction, yammer, 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 stimulates the muscles. And so when they fire action potentials, they're going to contract. And when there's no action potentials, no stimulation of the muscles, and they're going to relax. That makes sense? Yes, no, maybe?
Yes.
Excellent. Perfect. This area that causes this then, that fires the action potentials that stimulates these muscles, is what is known as the inspiratory center. Inspiratory center stimulates the muscle cells so that they contract, and when they contract, we inhale. So when it fires action potentials, that's what's going to happen. However, as we also mentioned, there is a second area. This second area is called the expiratory. Now, does it have to directly stimulate the muscles to get them to exhale?
No, since they're relaxing.
Yeah, no, we just want them to relax. So we're not going to stimulate the muscles directly. Instead, its job is going to be to inhibit the inspiratory center. Its job is going to be to inhibit the inspiratory center so that the inspiratory center does not fire. If this is firing all the time, our muscles firing all the time, our muscles contracted all the time, and that's a problem. But this one's job is to inhibit and I'll just put IC for inspiratory center there. But if it's constantly inhibiting the inspiratory center, then it's never firing action potential. We never contract those muscles and we never breathe. And so that's an issue. We need some way to cycle between these two. And the reason we're able to do this is because these two centers have, as I mentioned earlier, they have a circuit. And this special neural circuit is basically what happens is they have a reciprocal inhibition. And actually, I'm going to steal the space down here to write this. They have a reciprocal inhibition. Basically, what this means is they take turns inhibiting each other. They took turns firing and inhibiting each other. So actually, both of these form synapses with each other. And so his job is to inhibit the inspiratory center. The inspiratory center's job is to stimulate the muscles and to inhibit the expiratory center. All right. Now, there's one last thing we need to understand to see how this reciprocal inhibition works. This works because our inspiratory center and our expiratory center both have a special characteristic. Really two. Two special characteristics. The first special characteristic they have is the centers habituate. Anybody know what it means to habituate?
Delay?
Say again?
Delay?
Not so much a delay. Oh, I like that one. Get used to. Someone mentioned get used to,
Okay.
right? I like that. So kids are a perfect example, right? When you have that newborn baby, it just whimpers just a little bit and it gets your complete and undivided attention, right? You're right there focused on that baby, worried about its well-being and everything else about that, right? Whereas now, my two teenagers could be screaming bloody murder. Do I get worried and go running to see what it is? I'm not even aware of the noise, right? Maybe I have to turn up my earbuds a little bit, but I'm, they're sitting there, dad, 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 dad. Do I respond? No, I've completely habituated to them. The frequency of their voices, I have just been able to completely tune out. And that's what happens. The centers, plural, habituate. Basically what it means is they lose their reactivity to the cell. They lose that ability to respond. And when they lose that ability to respond, the other special characteristic of these cells is they have what we call this rebound effect. What this rebound effect is when the centers have been inhibited for a long time, they respond, they bounce back by depolarizing. Let's see how this works. All right. If we walk through the process, maybe we can make some sense of it. We will start here in our inspiratory center. Nope. Darn it. I didn't want that to be red. All right. Well, I'll change colors then. Great. Step one. Our respiratory center fires action potentials. When it fires action potentials, two things happen. It stimulates the muscles, and then of course our muscles contract. And when our muscles contract, we inhale. The second thing it does is it inhibits our expiratory center. So here we get a positive effect. Firing action potentials, and it's stimulated. Here we get an inhibitory effect. And when we get this inhibitory effect, our expiratory center is inhibited. And if it's inhibited, it cannot fire action potentials. All right. Simple enough?
Yes.
All right, excellent. But remember, what's going to happen next is that our expiratory center habituates. So what that means is it stops getting inhibited by the inspiratory center. The inspiratory center's chemical doesn't have a response anymore. It stops responding to it. And when it stops responding, it rebounds, depolarizes, and starts firing action potentials. And when it fires those action potentials, oops, undo that. Those action potentials inhibit the inspiratory center. And when it inhibits the entire the inspiratory center stops firing action potentials. And if it stops firing action potentials, it stops stimulating the muscles, and the muscles relax. So notice we've had an inhalation, and now we're getting an exhalation. However, is our inspiratory center going to stay inhibited forever?
No.
No, our inspiratory center now habituates. And when it habituates, it stops getting inhibited by the expiratory center, and it rebounds, depolarizes, and when it depolarizes, it starts firing action potentials. And when it stops and it starts firing action potentials, then guess what happens? It starts stimulating the muscle cell again to contract. It starts inhibiting the expiratory center again, and the process continues. Basically what happens is these two cells take turns firing their action potentials. The inspiratory goes first, stimulating the muscles, then the expiratory, causing the muscles to relax. Then it expires again, and then if the other one fires. And so by having this alternating circuit, this reciprocal inhibition, they are able to keep this nice pattern going back and forth, where we get a 2-second inhalation and a 3-second exhalation. So it's just that's the most simplest and basic of circuits. We use circuits like this for a lot of things that we do. Walking, all sorts of other type of locomotion type of activities, right? Even peristalsis, right? Where we have the alternating contractions of the circular and the longitudinal muscles. These types of circuits, this reciprocal inhibition, is something that is vitally important for many of the processes in the body. And here is one of the simplest examples of it here in our respiratory systems. All right. Questions on that? So notice, as we talked about, our ventral respiratory group is the part where we, oops, wrong button, is the part where we produce that eupnea. We generate that reciprocal inhibition cycle. However, as internal cues change, we can then modify this rate to make it faster, to make it slower, to make it deeper, to make it more shallow. But remember, there was a third region as well. And that third region is the area in the pons. Now, again, it's a respiratory group, lots of nuclei with lots of different controls. But primarily, what it does is basically two main things. One of the main things it does is it helps to fine-tune the respiratory center. where it helps to maintain the rhythm of that reciprocal inhibition so that it doesn't get out of control. In fact, if this area is damaged, one of the things that'll start to happen is you will start taking breaths that have longer and longer exhalations to it as it gets disrupted. So it helps to maintain that so that it's not jagged, it's not rough. It helps to smooth out and fine tune the rhythm. But the other thing it allows us to do is modify it based on more external stimuli. So for instance, if you're sleeping, right, again, one of the easiest ways to tell that your significant other is asleep and you can sneak out of bed to go play Xbox is by the change in the pattern of their sleep. When you're exercising, when you're eating, breathing can change. And then obviously I need that voluntary control of it to produce speech. Right? I need to control how rapidly, how fast air is coming in and out so that I can produce speech, or I can sing, or I can do any of those types of things. And, oops, the other thing that is related to our pontine respiratory group is our higher order reasoning and our limbic system. Right? When you are in pain, when you are aroused, right? When you are angry, do those things affect your respiration rate? To the part where you enthusiastically say yes to the easy questions that I'm asking. Yes, excellent, absolutely, right? Absolutely. So those are the other types of things that can affect it from the pontine group as well. Our limbic system, and again, our limbic system, we're also talking about our hypothalamus, can influence and modify those effects as well. Now, all of our areas are sensitive to our different types of stimuli. And as we talked about, the big three are carbon dioxide, pH, and oxygen levels. And I think this should be obvious, but let's do it anyway. The partial pressure of oxygen, when it increases, what does that do to our rate and our depth of breathing? Do we breathe faster or slower if partial pressure of oxygen goes up?
Faster.
Slower if it goes up. If our
Oh.
partial pressure and slower, and is it going to be more shallow or more deep?
Deep.
If we already have a lot of oxygen, do we need to be getting more air in?
No.
No, so it's going to be slower and it's going to be more shallow.
Okay.
Right? So maybe it isn't obvious. Maybe it's a good thing we're doing this. What about carbon dioxide? Okay, so well, and then here, let's do the opposite. If the partial pressure of oxygen decreases, if we have less oxygen, then what happens to our rate of breathing then?
It goes up.
Faster and
Deep.
deeper. Excellent. When CO2 increases, what happens to our rate of breathing?
Increases.
Yeah, faster and deeper. Excellent.
Shall.
Nope, faster and deeper. We want to get rid. We want to get rid of that CO2. So we take bigger, right? More ventilation, get rid of that air, bad air, right? Whereas if CO2 decreases, then slower and more shallow. And pH. As pH increases, and again, that one's the one that's always tricky for me, because remember, when it increases, that means it's getting less acidic. When it's getting less acidic, so really, if we think about pH increasing, we should really put over here with decreases. Well, actually, I don't know. I don't know which way it's more confusing. So let's just do it this way. pH increases. When pH increases and if blood becomes less acidic, what do you think happens to our rate of breathing?
Slower.
There you go. Slower and more shallow. And when it decreases, which of course means that it is more acidic, then we need to get rid of that acid. So it's going to be faster and deeper. Excellent. So the more metabolically active we are, the deeper and the faster our breathing is going to become. All right. Again, one of the things that you guys did, hopefully for your breathing exercises, was hyperventilate yourself. When you're hyperventilated yourself, what happened to how long you could hold your breath? Did it decrease when you're hyperventilated?
No, you can keep it longer, much longer.
Yeah, if it decreased, then you probably hadn't fully recovered from exercising yet, is probably what happened. When you hyperventilate, you blow off a bunch of excess CO2. So your CO2 level gets very, very low, meaning that your breathing rate is going to be slower and more shallow. It means you can typically hold your breath for a longer period of time as a result of that. So there you go. I think, so I'm glad we did take the time to go over that because I think it maybe wasn't as intuitive as I thought it would be, but that's excellent. Oh, and let's actually leave them there for a second. Of these three factors, and I think I've already given it away, but more importantly, your lab should have given it away. Which of these three factors is the most important in determining your rate of respiration?
CO2, increase of CO2.
Partial pressure of CO2, absolutely. All right. This is what you guys did when holding your breath. Notice when you're holding your breath, we talked about how you get a lot of oxygen into your blood. And remember, when you exercised and you were exercising more rapidly, even though your blood was going through your lungs more quickly, did your lungs have problem getting enough oxygen into your blood? No. Your blood isn't going to, remember, we saw how quickly that oxygen diffuses across. So your oxygen levels don't change. And when you hyperventilate, are you really getting more oxygen into your blood? Can you get more than 4 oxygen onto a hemoglobin?
No.
No. So notice whether you exercise, whether you're at rest, or whether you are hyperventilating, your oxygen levels stay the same. So if oxygen levels determined how long you could hold your breath for, when you were resting, when you were exercising, when you were hyperventilating, all of your holding your breath rates would have been the same. And is that what happened?
No.
No. So it's not that you hold your breath to a point where your oxygen gets so low that you're forced to breathe. It's the opposite. You have a critical CO2 level. Normally your CO2 level is here. You hold your breath while resting and it comes up. And when you reach that critical point, you have to breathe. When you exercise, you're producing a lot more CO2. And so you reach that point much more quickly. When you hyperventilate, you dramatically decrease your CO2 level, and it takes a lot longer for you to reach that critical point. So if you truly rested the way you should have in between the activities, you should have been able to hold your breath the longest when you hyperventilated, then resting. And how long were you able to hold your breath for when you exercised?
It was the least.
Yeah, did anybody make it past 30 seconds when they after exercising?
Not me.
Yeah, not too many people are able to, absolutely. Right, very few are able to do that because if you exercise vigorously, you're producing a lot of CO2. So absolutely the most important factor is CO2. All right. All right, excellent. So each of these factors are measured independently. And how are they measured? Where are they measured? Where do we take these measurements? How and where? Where do you think we measure CO2 levels and O2 levels and the pH of the blood? Excellent. We're going to use chemoreceptors. And where might those chemoreceptors be located?
In the alveoli?
No, it's not a bad guess. That would be a good place to have it, but that wouldn't necessarily be in the blood. Wasn't there some structures in some of the blood vessels that we talked about where we had clusters of chemoreceptors and osmoreceptors? Maybe monitoring the blood going to the body and the blood going to our brain. Where were those clusters of chemoreceptors located?
Aortic bodies and
Excellent.
um,
Aortic bodies, and there it is, the carotid bodies. Excellent. So those aortic bodies and carotid bodies are where we're measuring it, right? That's the blood basically being driven to the body. That's the blood going to the head. So we want to monitor the condition of that blood to see if we need to change the rate of breathing to make it appropriate. Excellent, 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 excellent. And here we see some of those pretty pictures. Here we see the carotid bodies, we see the aortic bodies receiving that information and bringing that information to the brain. Notice cranial nerve 10 is one of the primary ways that this is coming in. A 9 carries some of that sensory information as well. So again, these are the ways that we're bringing that information in. All right, and then we have these pretty flow charts from your textbook. And we did all of that. Excellent. But while those are the primary factors, as you guys mentioned earlier, there's some other factors that can change our rate of breathing as well. Your blood pressure. Typically, as your blood pressure goes up, what happens to your rate of breathing?
also goes up.
It goes up as well, right? Volume changes in your lung can affect it as well. Right. One of the things, obviously, as we talked about, when there's damage to the lungs and you're not getting as much air, you have to increase it. But here's another factor as well. You guys, again, didn't get a chance to use the spirometers in class, where you would fill your lungs to capacity, to as much as you could possibly get in there. When you do that, can you really get as much air as possible into your lungs? If I asked you to fill your lungs to your maximum capacity, and then I took a high pressure hose, would I be able to get more air into your lungs than you did?
Maybe not.
Maybe yes.
Yes.
Yes,
Maybe a little bit more.
Yeah, a little bit more. And that's smart. Why would you not want to be able to completely inflate your lungs as much as possible? When you fill a balloon for a party, do you put as much possible air as you can into it, filling it to its absolute maximal capacity? No, because it's much more likely to pop. And the same thing is true for our lungs. In fact, our lungs have a very special stretch receptor. This stretch receptor, basically, the more it stretches, the more it fires an action potential. Let's cheat and go back to this. We have a special, let's see, let's use orange, and I will sneak it in up here. A special stretch receptor that measures, this is supposed to be orange. And this special stretch receptor measures the stretch of the lungs. All right. Now, what happens is the more the lungs stretch, the more the stretch receptor fires action potentials. And it turns out what these special stretch receptors do is they inhibit the inspiratory center. So the more your lungs inflate, the more these stretch receptors send a signal to your inspiratory center saying, stop breathing in. I don't want my lungs to pop like a balloon. So the more they inflate, the stronger the inhibition, the more they can stop that inhalation. So you cannot fill your lungs to capacity because these special stretch receptors stop you from doing it, because if you were to do it, you'd likely damage your lungs. And this protective reflex, because that's ultimately what this protective reflex is actually called the Herring-Brewer reflex. Because good old Bob Herring and Bob Brewer did the research and discovered it and planted their flag in it. If you inhale dust or pepper or things along those lines, can that change your rate of breathing, causing you to sneeze, cough, hack, things along those lines? Absolutely. But other physical sensations, what about cold or heat? When you get warm, what happens to your respiration rate? It increases. So when you get cold, what happens to it? Decreases. This is one of the problems with some of those polar plunges, right? This winter equinox is right around the corner. In many places, Chicago is one of the most famous for them. On the winter equinox, many people will go out, cut a big hole in the ice and go jump into freezing cold water. Right? The polar plunge makes you manly or womanly or whatever it is that makes you. But one of the big concerns, one of the big worries about something like that is when you dramatically decrease your body temperature by jumping into that freezing cold water, that can actually, that big, huge decrease in temperature can cause a big, huge, massive decrease in respiration, and you can actually stop breathing as a result of it. Yeah, the shock to the body causes them to stop breathing, right? And like I said, all sorts of things involving our limbic system. It's one of the things that when they do with the, we talked about the lie detector test. Your emotions affect your physiology. So if you're angry or if you're anxious, your breathing rate is going to change. So they hook you up to a machine that monitors your breathing rate. And the inspector asks you your name. What's your favorite color? Do you like puppies? Where were you on the night of the 5th? Right? And that stress about that situation changes your respiration rate, changes your sweating, changes your heart rate. And those are the things that they're looking for on that lie detector test. One of those things they're looking for is a change in respiration rate, indicating that emotional stress. And then, of course, volition. What is volition? A big fancy word. Call up grandma. Hey, grandma, guess what? I have volition over my respiratory system. And she'll be very impressed and send you 20 bucks in the mail. Yeah, control, I like that. But more specifically, voluntary control. If I told you for 3 points of extra credit, you have to take 3 big, large breaths, would you be capable of doing that? Yeah, absolutely, right? Or 4 shallow breaths, or hold your breath and count to 10, right? That ability to control our rate of respiration is our volition. Now, are there limits? If someone's holding your head underwater, you're going to hold your breath, but are there limits for how long you can hold your breath for? Yeah, again, our levels of CO2 will override it, and eventually it'll force you to try to breathe, even though there ain't no air around to breathe. All right, that's one of the problems with drowning. Your volition can only go so far. All right, excellent. Here is the pretty picture kind of showing all these things going together, how they all affect these respiratory cycles and can modify our rate of respiration. One last big concept, well, one last little concept, and it's one we've already talked about. As I mentioned, all of us being healthy young adults, well, okay, maybe not young, but well, maybe not healthy. All of us being adults, what is our normal respiratory rate? All of you guys are young and healthy. I was talking merely about me. What did we say the normal respiration rate was as an adult? About 12 breaths per minute, absolutely. But what about that newborn baby in the crib? Is it breathing at a rate of 12 breaths per minute? Anyone heard a baby breathing before, been around a baby and they're breathing? It's creepy, right? What? No, it's way, way more. Absolutely, right? A newborn baby may take as many as 40 to 80 breaths per minute. Why so much?
Because of the size.
Yeah, they're tiny. They have tiny little lungs, tiny little bodies, and huge metabolisms, right? It's that potent combo of small lungs and massive metabolism, so massive oxygen needs. So not surprising, their respiration rate is much, much higher. It stays high through infancy, through age 5, but like I said, by the time we reach adults, we hit about a rate of about 12 to 15. But as we also talked about, as you move from adult to more elderly, often the respiration rate goes up again, right? Obviously, you don't get smaller when you age, well, it's not significantly smaller. But damage, loss of elasticity, weakness of muscles, all those things are factors that can modify, decrease in surfactant. All those things, as we talked about, are things that can decrease the overall volume of your lungs. And so as the lung volume goes down, as the efficiency of your respiratory mechanisms go down, you have to breathe more to get the same amount of oxygen into our blood. So it starts high, comes down, but then goes back high. Now, usually it doesn't get back as high as 40 to 80, right? If grandma's breathing 80 respirations per minute, then please take her to a doctor. But it does start to creep back up. All right. Questions on that? Excellent. Snuck it in at the end of time. That is everything that I wanted to talk about for today. So that is all of our big, heavy lifting regulatory stuff for our respiratory system. And so on Tuesday, when we come back, we will start the urinary system. All right. Although don't forget, you still have one more respiratory activity. You have that PhysioX that is due on Tuesday. But turn the corner, get that done, and start looking at the anatomy for the urinary system. Every question is important, so go ahead.
So you know how you said the Pons Respiratory Center, it influences our breathing during sleep?
Yes.
So I was thinking during sleep paralysis, like one of the things in sleep paralysis is you have trouble breathing. So is that the Pons affecting your breathing, or is that like psychological type issues?
That's a great question. My guess is that in those types of things, in the same way, it would be the same way, my guess, with like a nightmare and things along those lines as well, that would be more the stuff coming from the limbic system or from the hypothalamus, as opposed to but they may go through the Pons Respiratory Center. So it would, my guess is that ultimately it's coming from a higher area, but it would pass through the Pons respiratory group on its way. Absolutely. It just wouldn't start there. It would be probably much more associated with, well, like I said, nightmare, change, the changing in breath when you're having a stressful dream. That's definitely limbic. The paralysis one, my guess would be that it would probably be more hypothalamus, but honestly, I don't know if that's been studied. Again, not my area of expertise, so I don't know. But my guess would be, if I had to guess just off the top of my head, I would say probably hypothalamus. But the hypothalamus communicates with the pons, and then the pons communicates with the medulla. So yeah. Awesome. So that was a fun question. I liked it. All right. Any others? All right. You guys have a good weekend. Study hard, study well, be safe, and I will see you guys on Tuesday. Take care. Who knows? Maybe by then we'll even have a president. All right. See you guys.